A very good morning to all the participants. And it's a great pleasure for me to invite Dr. Venkatappa Rao, who is in the advisory committee of the Integrative Center for Research and Innovation in Biology, which is an autonomous scientific research institute established by a registered non-governmental organization named Braj Mohan Jha Science Research and Innovation Foundation based in Chandigarh. It was started as Indian Consortium for Research and Innovation in Biology with an objective to promote scientific knowledge spanning and integrating with all levels of biological organizations covering a wide range of research areas. Dr. Rao is an integral part of the institute and is in the advisory board. Sir received his postgraduate degree in zoology in first division from Ranchi University and PhD degree also from the same university in 1991 on the topic entitled Investigation on Possible Involvement of some Adrenocorticotropic Hormone in the Modulation of Adrenal Gland in Squirrel, Funambulus pinnati. He joined as lecturer in zoology at St. Xavier's College and promoted as lecturer in and is currently holding the position of associate professor. His teaching assignments and research include comparative anatomy, physiology, endocrinology, and reproductive biology. And he has authored and co-authored many scientific papers and books, and is also a part of. He is also an internal expert in the Department of Biotechnology, and he is in the Board of Studies, and also is a member to frame the reservation policy in the college. And he is also a coordinator of the self-study report of the college for NAC. And it's my great pleasure to welcome sir. And I request you, sir, to please come and speak a few words regarding our webinar.
Thank you, ma'am. I hope I'm audible.
Yes, sir.
Yes, sir.
Okay. Thank you. You have already introduced, it is my privilege that you have introduced me to this August event. You already mentioned that this Integrative Center for Research and Innovation in Biology is an autonomous scientific research institute. We abbreviated as ICRAB. And it is established under the Braj Mohan Jha Scientific Research and Innovation Foundation based at Chandigarh. See, we have this ICRAB having certain vision and mission. And the main our mission and vision is to spread the knowledge of biology, covering the wide range of research areas, including genetics, molecular biology, ecology, evolution. And our mission is also to promote the basic as well as advanced techniques in biology exploration and provide a platform for interdisciplinary research so as to promote the modern scientists to the world of research. The ICRAB embraces diversity and thrives on multiple perspectives to explore and achieve scientific excellence. It knows it values its responsibility to engage the communities and communicate the science and innovation. To pursue its vision and mission, the ICRAB is organized this webinar, the present webinar, on the GCMS data analysis and interpretation, which I believe will have a great impact on the emerging biologists. not to take much time, now I invite Dr. Pranav Bhaskar to deliver his valuable lecture. And I hope you will learn many new things from this webinar. Thank you. Thank you very much.
Thank you,
Pranav
sir.
Bhaskar.
Thank you, sir.
Thank you, ma'am.
Before Dr. Pranav takes over, I'd like to tell you a few things about him. He's a microbial molecular biologist and is having a research background in terpene cyclase enzyme and terpene biosynthesis from actinobacteria. He completed his PhD in biochemistry from the Department of Biochemistry, Punjab University, Chandigarh, and is currently working as a postdoctoral research associate in the Department of Pharmacology, School of Medicine, University of Virginia. His area of expertise is chromatography, spectroscopy, PCR, protein purification, HPLC, GCMS, electrophoresis, electrophysiology, Western blotting, northern and southern blotting, different immunological tests, RT-PCR, and so on. Now I request Dr. Pranav to please continue further.
Good morning, everyone. Here in Virginia, it's like night, and still, I think I got this energy to stay awake for this long so that I can tell you about GCMS, because I have that passion towards gas chromatography, mass spectrometry. Today, in this webinar, I'm not going to talk about the basics of GC. Rather, I will talk about the fundamental way how to analyze and interpret the data. I think this is the ghost that everybody is fearing of. Even when I started with GCMS, I was very much afraid of this data analysis part. And it was like kind of putting me down every time when I go for this, because I always felt that this is something which is not doable for me because I'm not a chemist. But believe me, you don't need to be a chemist. If you know what you are actually looking for and how you want to proceed with, you don't need to be a chemist. Let me share my screen, and then I will tell you that how we are going to proceed with this whole GCMS data analysis perspectives. So for this, I'm trying to connect. Just bear with me. Let me know if you can, if you are able to see the screen.
Yes, Dr. Pranav, it's visible.
Oh, okay, thank you.
And I would like to tell the participants that while he'll be explaining, if you have any questions, you can mention it in the chat box.
Yes, that would be great. And if you could put in your questions, and if you are able to do, means if you are feeling that this is a silly question, how can I ask to him? Then believe me, everything in science is just a silly question that we have to go behind it. Until and unless we go behind it, we are not going to resolve it. So let's go behind GCMS data interpretation today. Yeah, so today, the main motive is to make you aware of how to analyze the data, how to interpret the data that we get from GCMS analysis.
Dr. Pranav, have you started the recording?
Yeah, it is already. Yes, Dr. Venkat, आप कुछ पूछना चाह रहे हैं? Hello, Venkat sir?
नहीं नहीं, ये शायद दब गया है, सॉरी.
Oh, okay, no problem. So today we will learn what GCMS actually means and how we interpret the data that we get from GCMS. So let's start with it. Yeah, this is having some issue. Is the slide changing?
No, it's not changing.
Yeah, okay,
Yes, yeah, it's no.
fine. So, the whole webinar will move in this direction. First, we will talk about basics of introduction to GCMS, and then we will talk about instrument design, method and detector selection, then result interpretation, and in the last, and the most important, that how to present your GCMS data for your thesis or for your publications, for papers. So moving ahead with that, as you can see, this is a basic diagram of GCMS where you need a gas. That is why gas chromatography, you need a gas, carrier gas. Mostly it is helium when we talk about mass spectrometry or other like TCD, thermal conductive detectors. In that case, you use helium, but when you go for flame ionized detector, then you need hydrogen gas. And similarly, there are different uses. So we have different gases, like we use nitrogen as well. But for now, we will talk about the basics. So gas chromatography, mass spectrometry. Krishna, we will talk about it. So keep your interest, means keep active, keep your interest alive. And I hope I can make it understand how to calculate M by Z ratio. So normally, gas chromatography mass spectrometry is a hypernated technique. So what is hyphenated technique? This hyphenated doesn't mean that GC hyphen mass spectra. Hyphenated technique means when two analytical techniques are coupled, like a separation technique and a detection technique. Suppose in gas chromatography, there is a chromatographic technique for separation of the analytes, and the another technique is a spectroscopic technique to detect the fragments, to detect the ions, or to detect the analytes. When these two techniques are coupled, they are called hyphenated technique. So basically, GCMS does three things. One is qualitative determination, quantitative estimation, and to identify components in a mixture. So what is here? What are the basics for determining organic compounds? The first thing is purity. So in GCMS, we want separation in such a way that ultimately we get a pure compound. That is purity. The another one is stability. In GC, we cannot run any compound and means our analyte could not be anything. It could be something which can be volatilized, which can be stable at higher temperatures, which can deal with higher temperature without losing their general structural and chemical stability. Looking forward to the application of GCMS, we know that environmental chemists use GCMS for the identification of environmental pollutants in air, soil, water. The another one is food and nutrition science, It also includes health science, wherein uh, there are some sport uh, organizations like WADA, World Anti-Dope Agency. You would have heard about many athletes, they take drug to enhance their potential and to enhance their performance. So these drugs are detected by GCMS. So their blood and urine samples are taken and they were run on GCMS, and on the basis of the standards, they compare the peaks with the standards and they say, yes, this athlete had taken drugs before the race or before any sport events. Also, in food and nutrition science, for identifying adulterants in food, there are different chemical methods, but GC is one of the best technique to find the adulterants in um, food and beverages. The third one is renewable fuels. To identify natural renewable fuels from plants or like biodiesel, we use GCMS technique. The fourth one is forensic science. Again, the drug addiction and if there are any poison that had been given to some to the victim. So for these identification. But here I know everybody that we all are here are more interested in identifying pharmacological products or natural products. So this is the main motive. This is the main objective that we all are here to, because we all are working either in looking for phyto components or looking for natural products in different horizons of life, like in insects or in plants and bacteria and fungi, right? So this is the target. This is the actual target. So this is the basic that why we use GCMS, what are the applications, what basically GCMS does and what, how. Yes, Rahul, GCMS can be used for studying pheromones and other volatile compounds. Yes. So in case of in case of pheromones and also with other volatile compounds which are used in fragrant and hygiene companies, GCMS is very important, or you can say it's a very integral tool that we need for those studies. So when we talk about instrumental design, I'm in this part, I'm going to talk about how does a GCMS work correlating with the instrument design. So here, as you see, this is a gas cylinder. This will be containing inert carrier gas, that is a mobile phase. Here in the column, in this column oven, there you see a column having silica coated within, and that behaves as a stationary phase. And then this is mass spectrometer, that is detector, and this is uh, your computer wherein you get the chromatogram. But this is not so very simple. This works with a different kind of requirements, different kind of categorization, and the kind of columns we use, the kind of uh, sampler we use, the kind of sample we use, everything. Like usually what we do, we inject samples from either organic extract or aqueous extract or ethanolic extract, and we just put in through the auto sampler, or we inject manually, and then it does its job. We select the ion splitter, or you can say ionizer. We select the ionization program and then run the program, and according to the mass to charge ratio that Krishna asked, I will tell you how to calculate mass to charge ratio. So depending upon mass to charge ratio, this analyze, they get separated through mass analyzer and then finally detected on the mass detector. So in going into So going into these details, any question?
ఒక్క నిమిషం ఆగు.
Hello.
Yes, Dr. Pranab. I don't think
Yeah.
there are any questions.
Okay,
So
because
far.
yeah, okay. So let's move through the process. Let me walk you through the process. How does a GCMS instrument work? So there is a sample mixture that we already have, and we want to separate these analyte by the GC first. So there comes two different instrumentation. The first is for separation, and then another is for detection. So let's work with GC as a separator. So the sample mixture is first separated by the gas chromatogram before the analyte molecules are eluted into the MS detector. They're transported by this carrier gas, maybe helium or hydrogen, depending upon the use. If you are using flame ionized detector, then you need the sample to be heated under the flame to break into the ions, right? For that purpose, you need something which can burn, and that is hydrogen. That is why we use hydrogen as a carrier gas in a FID, in a GC FID detector, wherever FID is used as the detector. So the simple method is the sample mixture is transported by the carrier gas, which continuously flows through the GC column and into the MS, where it is evacuated by the vacuum system. So the sample is first introduced into the GC manually or by an auto sampler and enters into the carrier gas via the GC inlet. This is the GC inlet. If the sample is in the liquid form, yes, Dr. Rashmi, column is also different depending on the sample. Like if we are suppose it's not only the column, it's usually the column length, depend upon the sample type, not actually the column usually we use is silica columns, and it has like, suppose we are using C5 column, C8 column. So C5 and C8 columns are like, they have more affinity towards the analyte, and having 5 carbon as a what do you say, branch, or 8 carbon branch, depending upon that, the columns are different, but usually we use C5 column, and that is sufficient for having like most of the analyte we can analyze under a C5 column. Why you select C8 column? Because when you go for C8, C18 columns, because when there are, yes, C chain can vary. That is what I'm saying. We can have C8 column, and these C8 columns are used when there are when you are going to run your program at higher temperature, like you have to evaporate your compound at suppose 250 degrees centigrade, then C5 column is fine. But when you go beyond 280 or 320, then you need to have a column with more carbon branches for increasing the affinity, and also to hold the flow of helium gas, to maintain the helium gas flow as well within the column. For that reason, you have to maintain two things. One is the flow rate of the helium gas or the carrier gas, and second, the temperature. So to maintain these things, we can variate the length as well as the composition of the column. So when the sample is first introduced into the GC, it passes through the inlet and reaches to the GC column. And in this, there is an oven which heated the column, and depending upon their evaporation point, that you say as let's say boiling point, or you can say here evaporation point, wherein the compound started evaporating. So with the changes, slight changes in the temperature, like 20 degrees centigrade per minute or 15 degrees centigrade per minute, you are changing the temperature every minute. And with the changing temperature, whichsoever analyte become volatile at a particular temperature, it passes with the carrier gas to the mass spectrometer. I will show you a few examples how they separate out in the later slides. So now the sample components, or you can say the analytes, they are separated by their differences in partitioning or partitioning between the mobile phase and the liquid stationary phase. So here in the liquid stationary phase, it is itself the analyte in the column is the stationary phase, and the carrier gas is the mobile phase. So depending upon their partitioning and depending upon the temperature at which they get volatilized, they move, they get partitioned and they move with the carrier gas. Or for more volatile gases, their adsorption by a solid stationary phase, that too occurs. So in GCMS analysis, liquid stationary phase held within a narrow, like the tube's diameter is 0.1 to 0.25, and the length is only 10 to 30 meter. This is the most common column size that you use, that is 0.25 millimeter internal diameter and 10 to 30 meter length of the column. So after separation, like suppose you are going for high temperatures, then in that case, since you are moving with a slow pace, 20 degrees centigrade per minute, then you have to have a smaller column. 10 to 15 meter column will be fine. But when you are going with like 10 to 15 degrees centigrade and per minute, and you have like 250 to 80 degrees centigrade, then you can have a longer column so that you have a better resolved separation of the analytes. If you would have studied the plate numbers when you study HPLC columns or GC columns, so there are theoretical plates. So increasing the length increases number of theoretical plates, and that actually increases the better resolution or separation of the analytes. So after separation with GC, this so Dr. Jyoti, column is changeable. You can change the column. You can change silica columns every time when it chelates silica. When it starts chelating silica, you have to change the silica column. And also you can cut the size of the, means there are different sizes of column that we have, that we get, but you can cut the column into smaller sizes as well. If you see HPLC in there too, you cannot change the column in HPLC, but you can change the tubings to make it short so that you can have better movement or better mobility of your analyte into the HPLC column. Similarly, in GC also you can cut short the columns depending upon your requirement. But I think most of the general experiments, most of the general analysis of the analytes, 30 meter column is the common one, and it is fine. I have seen, I have used different kind of columns, and I have found that the column with 30 meter length and 0.5 millimeter external diameter and 0.25 millimeter internal diameter are good to go with any kind of analysis that you go for. Either you are using FID or TCD, thermal conductivity detector, or mass spectrometer. This works as a, you can say, as a star. So I have never found any difficulty with the columns until and unless it started leaching out, because if you don't consider the guidelines of that particular column, that you need not go beyond a particular temperature, then you have to sorry, then you have to see that if you are not going beyond that temperature and you are being in the limit, and if you are not using any acidic component in the column, then it is going to stay and it is going to live for like very long. But usually columns have their life cycle, and with uh, around 2000 to 3000 analysis, you will have to change the column depending upon the company that you use. I have seen Shimadzu is one of good company which makes the column, and their columns sustain a long, they have a long life. Yeah, Dr. Rashmi, your question was theoretical plates. So consider there is a consider this as a column. I don't have any diagram. So consider this as a column. And if the length of this column is suppose 30 meter or 10 meter per se. So you make, and you know that a particular, there is a small unit by which the analyte resolves. So suppose this is one, let me use a pointer. With the presentation pointer. Yeah. So wait a moment. Suppose there is this is nothing available. It's just a theoretical plate. Theoretical plate means you can easily consider that this is one unit from which an analyte can resolve, and an analyte can separate out. So for partitioning, there are number of you can just make an imaginary number, and you can see that if this is a column, and through this, if it passes through this and then to next, considering, and then it resolves in this much minute, suppose it resolves in 10 minutes, and there are number of theoretical plates that we have made like 100, and an analyte takes a whole 10 minutes to come out of it, then we can say there are 100 theoretical plates. But if a compound comes out in 5 minutes, then accordingly we can change the theoretical plates. We can say that maybe these two helps to resolve that particular analyte per minute. So considering that, you can, so this is just an imaginary plate wherein we can say that they resolve by this through this kind of a filter, that you can say these are imaginary filter plates through which the analyte resolves every minute. I hope it makes a sense. Yeah. So yeah. So after separation with GC, analyze doesn't require total baseline resolution unless the analytes are isomers. If they are isomers, then they might need baseline resolution. Otherwise, baseline resolution means they separate from each other in a way that they can be easily identified. That is baseline resolution. So until and unless they are isomers, this is not required. But if there are different analytes in a particular mix, then it is going to separate out depending upon their temperature of volatility. As they reach their temperature of volatility, they get volatile. And suppose there are A, B, C, D, E, 5 analytes in the sample mix. So analyte A, suppose it volatilized at 100 degrees centigrade. Analyte B separates at 120 degrees centigrade. And similarly, after every 20 minutes, they separate out. And our program is like after after reaching 100 or 80 degrees centigrade, for the column temperature, it start increasing the temperature by 20 degrees centigrade per minute. Then what will happen? As it reaches 100 degrees centigrade, analyte A get volatilized and comes into carrier gas and moves out. After again next 20 minutes or next 20 degrees centigrade or the next minute, analyte B get evaporated, vaporized, and it get uh, carried away with carrier gas. And similarly, C, D, and E. So this is how they are going to resolve automatically. So there we don't require a baseline resolution. But if there are two analytes and both of them are isomers, yes, there can be more than one analyte at the same temperature. That is what I'm saying. Mostly the isomers can come at same temperature, but then you have to have a baseline resolution. For that, then you have to have tandem mass spectroscopy or time of flight mass spectroscopy attached with your GC. So that will be GC MSMS or GC MSTOF, in which one analyte will be hold, because there are two analytes, they are isomers, so they will be broken down separately. They will be ionized separately in different um, detector, mass analyzer. So that I will be talking later on about that too. Yeah. So until and unless the analytes are isomers, they are not required to be separated on the baseline. Yes, Dr. Jyoti. Please
Yes,
ask your
sir.
question.
Yes, yes. Sir, actually, when the two samples they are having the same the isomers, they are actually volatilizing on the same temperature, but
Yeah.
their rate of separation would differ according to the M by Z ratio. Since they are traveling a long time in a column, so
Mhm.
by the time they reach to the detector, there will be the difference, isn't so?
Uh, See, for isomer, the mass by charge ratio remains same. It is not going to differ much. Or it is not going to differ because we ionize them on the basis of their. We have different ionizers. Suppose we are using ESI, electron spray ionization. So the
Electrospray analyzer. Yeah.
yeah, or EI, electron ionization. So in either of the method, what will happen? Or if we are using chemical method of ionization, so there will be they are not breaking down, actually they are getting one electron or releasing one electron. So the difference of the charge will be one, either in suppose there are two isomers and we say E and S isomers, right? So suppose we have E and S isomers or any structural isomers, suppose we can say ethyl alcohol, or you can say propanol and um, ethanaldehyde. There are two, these are two isomers, right? They are functional isomers. So what will happen? They will break accordingly. Suppose if we are talking about C2H5CHO, then it will break as CS3, CS2CHO. So there will be CS3 plus 1, and the another will be CS2CHO plus. Oh, sorry, CS2CHO minus. So there, the ratio of the difference of their charges is just plus 1. So it is not going to make difference. So they get separated on the basis of the quadrupole that is used in the mass spectrometer. So there can be single mass, single quadrupole, double quadrupole, triple quadrupole, or you can say MS that is coupled after GC. So that makes the actual difference. In those, uh, depending upon their charge, if it is positively charged, then what will happen? In the quadrupole, we provide the magnetic property in such a way that it deflect the positive charge to one direction and the another to another direction. And there are again one mass analyzer wherein, depending upon their charge, it deflect that to the uh, vacuum pump, wherein it got vacuum and hit the detector, and then we get the chromatogram. So that depend upon MSMS or the tandem. Okay, thank you. So where I was, okay. Yeah, so I was telling that normally the resolution of the analyte doesn't require baseline resolution until and unless it's the analyte is an isomer. The neutral molecules elute through a heated transfer line that I already told you. Depending upon, this is the heated transfer line, and depending upon the temperature variations every minute, the analyte get vaporized and carried away with carrier gas. Now the method and detector selection. Oh no, we are still not over with this because mass detector and everything is here. So within the mass spectrometer, the neutral molecules are first ionized, most commonly by electron ionization. We already talked about it when Dr. Jyoti asked the question. In EI, basically an electron produced by a filament is accelerated with either 60 electron volt or 70 electron volt, depending upon the kind of analysis you're going to do. And that actually provides a force to that electron, and that filament so that filament, when produce an electron, that electron knocks an electron out of the molecule to produce a molecular ion that is a radical cation. This high energy ionization can result in an unstable molecule, or we can't say it as a molecule, it as a molecular ion, and excess energy can be lost through fragmentation. Bond breakage can happen, due to which loss of a radical or neutral molecule and molecular rearrangement can also occur. This all result in a sometimes very large number of ions of different masses, the heaviest being the molecular ion that you will see at the that you see at the last of the mass spectra, and the heaviest being the molecular ion with fragment ions of various lower masses that we say for a specific analyte as a fragment pattern or ionic pattern, right? So there you by that you can find or by depending upon different structure of the analyte or molecular formula of the analyte or bond breakage, how bond breakage has occurred, what is which part of that analyte has retained the charge. Depending upon all these things, you get the peak pattern. So the longest and the heaviest being the molecular ion. The next step here is to separate the ions of different masses, which is achieved based on their mass to charge by the mass analyzer. Now the mass to charge ratio comes over here. Depending upon the mass means heaviest. Suppose if there is a heaviest ion, it is going to separate even if their they vaporize at a early temperature, which is unlikely possible, because if the formula mass is high and if there are number of bonds, then there are very little chance that the heaviest molecular ion or the ion peak comes first. It will definitely separate at the end. So depending upon the formula mass, that is why mass to charge ratio. Charge is always +1 -1 in case of mass spectra, normally what we use. Even if it is FID, there also mass to charge ratio, the charge is +or -1 or M+1, whatsoever is the molecular ion, just +1 or -1 will be the charge. So here in mass spectrometer, there are different mass analyzers, and that we will talk here. So when we go for method and detector selection, first we will talk about method and detectors. So there are two types of detectors. One is called destructive gas chromatography detectors. Destructive means here, can any one of you guess why it is called destructive gas chromatography detector and non-destructive gas chromatography detector? I would like to know what your idea about destructive and non-destructive. Yes, Dr. Rashmi.
Is it so because it is breaking the bonds?
No, this is not the reason. Yeah, because either it's destructive gas chromatography detector or non-destructive, both are breaking the bonds. Both are breaking the knocking out the electrons. Yeah. So no.
I guess because in destructive we cannot recover the analytes.
Perfect.
So.
Yes. Yes. So in destructive gas chromatography, what happens? The compound or the analyte you cannot recover back because it is either going to be you can say that in this the column effluent are transformed, burned or mixed. For example, if you want to recover, it is very impossible, but in non-destructive it is possible to recover. So you can recover your sample and do other experiments if you are using non-destructive gas chromatography detectors. But we are unfortunate that most of our application falls in destructive gas chromatography detector category. For example, flame ionization detector. So in flame ionization detectors, what happens? We heat, or rather we burn the analyte using a hydrogen flame. And here we use hydrogen gas as a carrier. So a component within the analyte will then become ionized and lose an electron when they get burned. That makes the flame more electrically conductive, triggering a measurable signal for the detector. Another is flame photometric detector. In flame photometric detector, similar to flame ionization detector, it involves burning compounds in a flame. And in this case, a photomultiplier tube is then used to detect spectral lines as specific elements emit light. So when you burn specific compounds, they emit light. And those lights are photomultiplied in a photomultiplier tube, which are detected as spectral lines on the detector or on a photographic plate. The third one is nitrogen phosphorus detector. In this, obviously, you are going to use a nitrogen gas as a carrier gas. And also, when we use hydrogen, we have to run a parallel carrier gas, that is nitrogen, to cool down the column. We are just using the flame and flame for burning the component within the inlet, not beyond that. After that, it is going to burn the column and you're not going to get any separation. For that, after the components are burnt in an inlet with the help of flame ionization detector, with the help of flame ionizer, then a separate line of nitrogen gas flows with hydrogen gas, maintaining the temperature so that it do not affect the column property. So in nitrogen phosphorus detector, it is especially designed to selectively detect the two elements for the namesake based on the way they alter the function of metal bead. That's it. It is not very commonly used, nitrogen phosphorus detector. It is commonly used for in environmental pollution detection, but nowadays mass spectro, FID, these are the common methods. More than that, if you go, then there is one non-destructive, that is electron capture detector and photoionization detector. These are used. After nitrogen phosphorus detector, there is one atomic emission detector. Atomic emission detector, in this, the sample enters into a chamber after eluting from the column, wherein these wherein the samples are energized to induce a plasma, energized with the help of atomic emission, which itself causes the sample to decompose. So whenever you are using any emissive component, which can emit beta or gamma radiations or those kind of things, wherein radioactive compounds are used, in that case, you can use atomic emission detector. So which causes this, when it elutes from the column, the sample enters into a different chamber in which they get energized with this atomic emission, inducing a plasma, which causes the sample to decompose. And this creates an atomic spectra from certain elements, which is then diffracted and detected. So in the previous one, where flame photometer detector was used, there was a photomultiplier tube giving a spectral lines as a specific elements emit light in the visible region. But here in atomic emission detector, there is a atomic emission spectra that you get from certain elements, which is then diffracted and detected on the last one, which is of our concern, is mass spectrometer. So this is one of the most common detectors. Mass spectrometer is used to identify analytes based on their mass spectrum. It can also be used with liquid chromatography. It can also be used with addition of time of flight or mass. So they have different combination that can be used for different purposes. Now we are going to talk about non-destructive gas chromatography detectors, and these are thermal conductivity detector, also known as catharometer. So catharometer spelling is Katharometer. So this thermal conductivity detector measures changes in thermal conductivity of the signal, thermal conductivity of the compound as a signal to detect compounds within an analyte. So suppose if you are using a compound and you have broken down the compound into smaller fragments or smaller analytes, then you need to detect them, but you don't have any other way. So then what you will do, you will find the value of K for each analyte, for each compound, with the help of their thermal conductivity, with the help of the changes in their thermal conductivity. So suppose there is a compound A which has certain thermal conductivity because of there are n number of atoms involved. As it breaks down into two, B and C, their number of atoms changes, so their conductive property also changes. And on that basis, it can be detected. Then another one is electron capture detector, ECD. So electron capture detector detect molecules in gas by attaching electrons to an analyte molecule. So here, what happened, ECD, here the compound attracts and captures an electron. And the change in the current with the detector chamber, because if electron is flowing, there is a flow of current. So depending upon the change in current with the detector chamber, the molecules are detected. So every molecule have their own electrical conductivity. So as the electron attaches, there is a change in their electrical conductivity, and on that basis, their proportional signals are detected for the analyte concentration. So with different varying concentration, if you are adding 2 micromolar of A and 2 micromolar of B, it is easily detectable if they are in the range of electrical conductivity. But if they are having multiplication of conductivity, suppose A has double the conductivity of B, then in that case, if you are having A twice its initial concentration, then A and B both are going to give you the same signal. So for that, it is not possible to get the signal. So for that, you need to have same concentration of both the analyte. Then one is photoionization detector. This photoionization detector, they are also, they should be vaporized, and gases are measured instantaneously. So in photoionization detector, gases mainly are measured, and these are used for pollution. Like whenever you see that SO2 gas is high in a particular city, so photoionization detectors are there. So you don't need to have a GCMS system, just a photoionization detector with a small gas collector, which collects the overhead gas. It is also called head space gas collector or head space chamber. So head space chamber attached with photoionization detector can give you the um, concentration of particular gas within the environment. So these are typically used for volatile organic compounds. The detector uses UV rays to ionize molecules, causing them to release an electron and form a positive ion. So whenever we see SO2 concentration, they were given in ppm, parts per million. So what happens when an electron releases, the analyte forms a positive ion, and that's turned into a parts per million meter reading when electrodes collect those ions. So as the ions, positive ions, hit the electrode as many times, it counts, and it give you a count in parts per million, ppm. And the last one is olfactometric detector. So for this detection method, trained humans are used to smell gases. So this is not a kind of detector, this is kind of a human intervention. So like you would have heard about, there are tasters in certain industries, like wine tasters. They are being paid hugely. If you have a good taste buds, and you can taste any beverage, you are going to be one of the highly paid uh, human being on the earth. So they are similarly, there are olfactometric detectors. They are humans. They smell gases and provide information about the presence of certain others. So these are, again, this is just a kind of detector that we are talking about. So nowadays we have AI based olfactometric detectors. So nowadays we are not using humans for detect these gases, but there is a system which convert these smells into electrical signals, which is detected by AI. They are like AI system which detect these. Now the another one is mass analyzer. The first one that we talk about is detector. Another one is mass analyzer. So mass analyzers are of two types. One is unit mass resolution that we talked about, quadrupole one. The another one is ion trap. We will talk about it. The another one is high mass resolution. And this is an advanced technique or the advanced method of detecting the masses called high mass resolution, or HRMS. So nowadays, GC is connected with HRMS. HRMS is nothing special. It is just a time of flight, or having magnetic sector, or orbitrap, or mass, double quadrupole, triple quadrupole, tetra quadrupole. People use different quadrupoles along with time of flight to give you more sensitive detection. That is called high mass resolution. And you need more sensitivity for the detection when your detector system is not like up to 0 to 500 masses. When you go for higher masses, there you need this kind of time of flight and magnetic sector, orbitrap. So there are numerous different mass analyzer types, and this is where the vast differences in the mass resolution comes. So, and as sensitive the instrument is, higher its cost. So whenever you talk about unit mass resolution, in comparison to unit mass resolution, high mass resolution is like twice or thrice in the price. The price is like 3 times of unit mass resolution. So mass resolution is the ability of the mass analyzer to separate ions with very small difference in their mass to charge ratio. Unit mass resolution instrument, when you talk about unit mass resolution instruments, they can only separate nominal masses or those down to a single decimal place, whereas high mass resolution can separate them to 4 or 5 decimal places. So normally what we are using in our universities or in a small setup of where we are doing mass, we are using unit mass resolution instruments. But when you go to industry, there they use high mass resolution instruments. The most common type of unit mass instrument is quadrupole. So as you can see, quadru means 4, poles means poles, and depending upon their rotation, they are parallel, oppositely placed positive and negative terminal of poles, and they rotate in X, Y, Z, all the 3 directions. And depending upon their rotation, whatsoever ion is coming through the ion source is changes. If there is positive, the ion here will go and combine with the, if there is a negative ion, that negative ion binds to the, or make a bond with the positive ion, or attaches with the positive ion, attracted towards the positive ion. Oh, sorry, positive quadrupole. So it will move towards the rest, which are not attracted, they get deflected and reach to the ion detector. So by that, you can decide that you are going to do positive mass spectrometry, or means +1, positive ion spectrometry, or negative ion spectrometry. So depend upon the quadrupole and the moment that you provide to the quadrupole. So it works in a 2 mode. One is full scan mode, the another one is selected ion monitoring mode. In full scan mode, where all ions are acquired across a mass range, normally we do full scan mode. We run the sample and we give the range like 50 to 500, and whatever we get is a mix of different RT values, and at each RT value there are different analytes at different data points. I will show what data point is, how data point means how we can say that this data point have these spectral lines, this data point has this spectral points. So, and each data point indicate a retention time. Now, if anybody can tell me what is the difference between the retention time of HPLC or liquid chromatography and retention time of gas chromatography? Can anybody tell me the difference, so that I can understand that you guys know what is the basic difference between the retention time? I can give you a hint. In gas chromatography, this retention time is mainly due to the temperature, and so we can also call it as retention temperature.
So the time taken by the analyte to come out from the column in HPLC, that is calculated as retention time.
Yeah.
The time
And
for
in
the
gas?
analyte.
Yeah.
Yeah, in gas also, when it is actually flowing with the gas, since the carrier gas is the mobile phase.
Mhm.
So then when the analyte which is coming to the detector,
Mhm.
and then calculated by the M/Z ratio, that could be the retention time.
You can say that, but to understand it, I give you a hint that you can call it as retention temperature as well. So this
Yeah,
RT
yeah,
value,
exactly. It's
yes,
the retention temperature. That's what I'm saying, because it is moving along with the temperature gradient, which is through the
yes,
column. Yeah,
The term
and
that you use,
because of the
temperature
temperature
gradient,
gradient, that is
yes,
yeah,
yes, the term temperature gradient that you said is the
yes,
actual catch. Yeah, Dr.
yeah.
Rashmi, you are saying something.
What I feel is that in HPLC, as far as
Mhm.
I know, it is the time it spends in the column.
Yes.
Time it remains in the column, whereas in the gas
Uh.
chromatography, the time it takes to come out of the column and strike the detector, I think, if it would be the
I
right
think
word.
both the thing retention in the column and coming out of the time taking to come out of the column is the same thing. But
Yeah,
you
essentially,
you yeah.
we can say, but what is there, since we get a peak on the basis of peak of the analyte on the basis of retention time, and the
Mhm.
peak will come out only when it reaches to the detector, right?
Yeah.
So that's why we say, in general, what we define as retention time is the time taken by the analyte to actually come out from the column, based
Yeah.
on any parameters, based on their adsorption, based upon their many other parameters.
Yes.
Because it will strike when it will come out.
Yes, definitely. So these are more or less same kind of thing, but if we have to explain it, we need to use these terms: concentration gradient in terms of liquid chromatography and
Yes,
temperature gradient in terms
temperature
of gas
gradient.
chromatography. So
Yeah.
how you define that in concentration, when you talk about retention time in liquid chromatography, you say that suppose you have a different concentration of A and B. You when you do HPLC, you always run A and B, two mobile phases in a ratio, and you mix it, and then it moves within the column. So what happened, as A is a neutral thing, as B increases in ratio, the analyte start eluting out, and the time it takes from the first minute till it comes out of the column, the time it retained in it, and add the concentration gradient at the particular concentration of B, it elutes out. That whole time depends upon the concentration gradient, and that is the retention time for that particular analyte. But in GC, you cannot say concentration gradient, you say temperature gradient. I gave you an example in the initial that we have 5 analytes, A, B, C, D, and E. So at 80 degrees centigrade, the program changes to 20 degrees centigrade per minute. And the analyte A comes out at the first minute, because it reaches to the temperature 100 degree, and 100 degree is the vaporization temperature for analyte A. So what will happen, you will have to calculate the whole time, like you heated the oven till 80 degrees centigrade in 6 minutes, and then in the 7th minute the analyte got vaporized and came out of the with the carrier gas and hit the detector. So this 7 or 7.12 kind of, you will see that whenever you get the GC data, you see the RT values as 7.12 or 13 or to the decimal places. So that 7.12 is the total time, retention time of that analyte A. Yes, Dr. Rashmi.
I have a very basic question here, that
Yeah, sure.
we find that retention time means, first of all, means the question will be in two parts. So the
Mhm.
first part of the question is, does retention time of the analyte vary, or it is same for the same? And why does it vary in different, means I have seen several papers in which the retention time was varying. Why
Yeah.
was it so?
So retention time depends upon the length of the column, first thing. It also depends upon the ionization program. Suppose if you are using 70 electron volt and I use 60 electron volt for ionization, the retention time of the analyte changes. If you are using flame ionized detector and if you are using flame ionization, in that, the temperature at which you are burning the analyte and the length of the column, it depends upon both. So It varies
So can we
for
say that,
Hmm.
I'm so sorry. So retention time cannot be used for the identification of the compound or the
No.
analyte?
No, no. If you are using the same machine, suppose, same column, same program, and you have a standard, and then you run, and then you see at, suppose you got a, like in this, I'm taking an example, just suppose it came at like 5.2 minute, okay? This is your standard, which came at 5.2. And similarly, you got a peak, smaller or bigger, doesn't matter. You got a peak, which is again at 5.2 minutes. And when you uh, tracked its mass spectra and you find the similarity, then yeah, on the same program, same column, same machine, you can have same retention time for same component. But the same component, even if you have a standard, you run on different machines, you will get different retention times. Different column length, you will get different retention times. Yeah. So this is not a parameter that we can rely on, but yes, it gives you an idea. Suppose but yeah, one more thing, even if you change the length of the column or program, it is not going to be like a huge difference between the retention time. Suppose if I have analyte A and I ran it on one machine and I saw it at 10.38, suppose, and the another on another machine or another column, I saw it at 11.2. That could be +1, -1 in the will be the difference in minutes, not huge difference. You are not going to see that. It's not like in one, the same sample gives you at 11 and in another you got it at 20. It could not be possible.
I have seen in few papers for the
Mhm.
same palmitic acid, they have mentioned around 19 in few papers, and in some they have mentioned 25.
Mostly when you go for FID, so it depends upon at what temperature you are burning palmitic acid, means you are burning the components. So there, so think it like the way that it's when you get the fragments, your detector actually collects the bits and pieces and then analyze it. So sometimes at when you burn at like high temperature, you can get to see this big difference between the RT values. But normally in mass spectra, you don't see this huge gap. And palmitic acid or normally whatsoever the lipid profile you go for, they are usually done on FID. They are
Yes, sir.
detected.
I think it is more RT value would be more relevant for HPLC analysis rather than
Yes,
GCMS.
yes, yes,
Yeah.
yes.
And
For
the
GCMS,
students
you
who
will
have
have to
joined
go.
this, yeah,
Uh-huh.
the students who has joined this webinar need to be clear about this concept that
Mhm.
while running with this standard, because they do it in their regular basis in our
Yeah,
laboratories.
yeah,
So by comparing with the any standards, the RT value would be same if we use the same machine and keeping
yeah,
everything constant, and that
yes,
should be clear to them.
yeah.
Yes, sir.
So this is a very important thing to understand when we run a standard. See, if we are running a scan mode, we don't know the analyte what we are looking for, then it is quite difficult to say that RT values will give you a kind of, because you don't know what standards you are going to use. You don't have any idea. Suppose we are using any unrelated native compound, mix of sample, then what will happen, you don't know what is the component, so you have to go for a scan mode and you don't know the standard or anything, so this is very difficult to go. Then you have to go with NIST library. So depending upon the mass spectra, you go ahead. RT value is only helpful in GC when you are running it on sim mode and you already have a standard to compare with the same program, with the same component, with the same everything on the same machine, same column length. If you have a standard, then you will run sim mode. I will tell you what sim mode is. It's not a mode that you, it's not a SIM card that you use in a phone or any machine. It's a, it's a selected ion monitoring mode. So in this, you know that what you are looking for. So in that case, you can use the standard and you can have same retention time. But yes, Dr. Jyoti, it's true that in GC, retention time is not a good parameter to look for. You always have to go with the spectra that you get, the mass spectra. And again, I make it very clear that even if you are using GCMS, it's a very sophisticated technique, but it is not a confirmatory, it is just a, what you can say, analytical technique wherein you cannot be sure enough that what you have got. You can only be sure after doing further experiments like NMR and other. GCMS can just give you an idea that what you are dealing with. That's it. So yeah, coming back to the scan mode and SIM mode. So scan mode is where all ions are acquired across a mass range. You give a mass range that I need to, for example, when you go with your sample and you give it to the technician who is running the GC, he asks that what you are, or you have a form in your institute wherein you have to mention that I have to, the mass should fall between 50 to 500, right? Or you go there and they ask you that what is the mass range that you are looking for, and you say 50 to 500. Or the common method is that if you don't say, they run between 50 to 500, right? So this can only be done in full scan mode, where all ions are acquired across a mass range. And this is useful for identification of unknowns, method development, or for doing qualitative and quantitative analysis for higher concentration analytes. But when you go for selected ion monitoring or SIM mode, where only selected ions that represent the target compound, that like here, you know that I need a compound, something which is going to be this, then only it will be detected. And then when you go for mass spectra, depending and comparing your spectral lines, you can say, okay, this is the compound that we have targeted and we got it. So this is basically useful for trace analysis or higher, when you work at a higher sensitivity, where you require higher sensitivity, then you use SIM mode. When you already know what is the target compound, you just need it to be very clear that it is not contaminated. It is having, means you, when you want to trace the compound, that it is not contaminated and it is highly pure, and you are further going to use this compound for your analysis. So here we have seen quadrupole, ion trap. So what is ion trap? Ion trap is also a scanning instrument, but is three-dimensional. It traps the ion in mass-dependent orbits before ejecting them sequentially to reach the ion detector. Yeah, so it's actually traps the ion. It's kind of ECD, wherein electrons are captured. So here ions are trapped in a kind of chamber, three-dimensional, and it depending upon the mass, and it orbits around the you can say analyte, and before ejecting them sequentially to the ion detector. The third one is time of flight, that I have not talked about, sorry. Yeah. In high mass resolution, there is time of flight. So time of flight mass analyzers, they separate the ions based on the time they take to travel down the flight tube. And there are again, like we have quadrupoles. So there are quadrupoles, so they deflect. They have large tube, means time of flight. For that, there is a large tube-like structure. It's not this as I'm showing. It's a it takes like half of the room. So it's a big machine, and there the lute, it passes through different quadrupoles and it takes a long time. So depending upon their mass they get, and depending upon their with the same kinetic energy, The lower mass have a higher velocity and they arrive first, and the one with higher mass, it has low velocity, so it will come later. So depending upon the time of flight, depending upon the kinetic, the mass, and with the same kinetic energy, it separates the analytes. So it can take around, the acquisition rate of time of flight is around 1,000 spectra per second, and that is huge. You can have like, if you collect the data in your hard disk, then running 10 sample can fill 1 terabyte of data. So you can imagine that how many spectra per second they are generating. So time of flight takes like a lot of data in your computer, a lot of space. Then the another HRMS instrument is magnetic sector. So magnetic sector mass analyzer, it basically bends the trajectory of the ions. It basically deflects the ions to separate them using electric and magnetic fields together. So magnetic sector GCMS instruments are more commonly found in where you are working with isotopes. So there you can find magnetic sector GCMS. Then the third one is Orbitrap. So Orbitrap, you can say that the ions orbit around a central spindle-like, means a central spindle that has in that machine, and with a definite frequency, and they move up and down the central spindle in mass to charge dependent ratio. So if there is a difference in mass to charge, then the one with heavier mass will move down and the one with lighter mass will stay up. And on the basis of their movement, or on the basis of their orbital movement, they are separated. So now we are going back to that diagram. Yeah. So after the ions have been separated by the mass analyzer based on their mass to charge, they reach the ion detector, where this is the ion detector. So it reaches the ion detector where the signal is amplified by electron multiplier. For most low resolution mass spectra, there is an electron multiplier. For HRMS, there are multi-channel plate multipliers. So they are used. So you can simply say that either it is having single multiplier or single plate in a low resolution mass spectra, and with high resolution mass spectra, you have multiple plates to resolve them. So for for amplification and multiplication of the electrons, the signal is recorded by the acquisition software on your computer. There are in different formats with different, like if you are using Thermo or Waters or Verizon, so with different or Agilent, with different manufacturers, they have different softwares. And I have used Thermo Fisher and Verizon and Agilent, and I have found that more or less all are same. If you know the fundamentals of GCMS, you can use all the software. They are not that difficult to handle. Yeah, so we talked about mass analyzers, and here in scan mode, there are like to understand this. So this is just the quadrupole system. It talks about the dual acquisition. So whenever you have these are the sizes of the molecule, you can see. So if you have a bigger mass, but you are not very much concerned about the stability, then you can use 4 quadrupole, which have smaller passes between the quadrupoles, and with the change in the charges or the electrical conductivity or the magnetic field, the different charged molecular ion passes through it. So mostly the smaller with the charge they pass, or the bigger with no charge or with charge, even they get deflected to the another quadrupole, another pole. There are collision cell wherein there are 6 different rods, and they are like basically they're basically kind of colliding to each other, so that they generate energy, kinetic energy, and that moves the ions through it. So they provide the kinetic energy to the analytes. And the last one is analyze multiple product ions in sim mode. So when you go for, what is the target molecule, then you can go for multiple product ions, wherein a single analyte can, when you see that, what was that? Sorry, I think I missed it. There it is. Yeah, in sim mode, as you get this RT values, when you go for a spectral analysis, you can see there are, it can be 5.2 here. There is an analyte moving at the top. There is suppose 5.15 or 5.35 is the tip here. So here the you will see different spectral patterns, and again at this value, at this end where it's suppose 5.4, you will see different spectral lines. So by sim mode, you can find different spectral patterns with different analytes coming out at the same peak. So here you can say, here you cannot see the peak tailing, you can see a resolved peak here. But in scan mode, you can see the peak tailing, wherein more than one analytes are merged, and they are coming after, like in every 2 second, 3 second, 5 second, you can see many analytes coming out at different RTs, but they're very close, they're not resolving very well. So you can find that difficulty in a scan mode, but not in sim mode, because you already know the target molecular ion. Yeah, so now we are going to talk about GC-MS-MS. So we were talking about tandem, right? So GC-MS-MS is nowadays more commonly used. This GC-MS-MS in wherein you use more than one mass spectra, more than one quadrupole. So there you can see one mass to charge separation and another mass to charge separation. So these are two mass analyzer or mass separator, and that is why it is called tandem. Tandem is they are coupled one after another. So if there are, when you say, you would have heard about the term triple quadrupole. So triple quadrupole means one quadrupole is one mass separator. So triple quadrupole means 3 mass separator aligned one after another, and they have different orientation, and they have different magnetic field, electrical and magnetic properties. So they deflect different charged ions. As you can see here, when the sample get ionized in GC, it has some positive, some negatively charged. So the first mass to charge, it gives you the precursor ion. So the question is, what is precursor ion? So during the first mass analyzer selects the ion called the precursor ion, and this is the first one which is going to give you the final, what do you say, molecular ion. So this is the precursor ion. So this precursor ion is further fragmented. So you got one big ion, that is your analyte that you want. It get broken down into different fragments again, and they are again separated by MS, second mass spectrometer. And so you get different fragment patterns. That is why when you use single quadrupole MS and you use double quadrupole and triple quadrupole, you see that a single analyte, the same analyte that you have used in all of them, you see more number of spectral lines. In first, you will see less. In second, you see more. And in third machine, you will see furthermore. So it's like, suppose if you have broken down a analyte in single quadrupole, you get like 100 spectral lines. But when you use the same, when you take that a precursor ion and you move into the second analyzer, it breaks down into further 200 or 500 spectra, and then on the third you get like thousands of. So according to that, you select the relative intensity of the ions that I'm going to talk about next. Okay, then the next is result interpretation. So we are now, I will stop here, and I want to know if you all have any queries to ask in this area or till here, because result interpretation will be more going to be complex, but it will be, I will try to make it as simple as possible. So yeah. So if you guys have any question till method and detector selection, please let me know. I think there is no question. Yeah. Go ahead.
Pranav,
Yes, sir.
actually, I wanted to tell all the participants from my side
Yeah.
that techniques play a very important role in innovative research. From the society side or from the organizing committee, I always wish that the participants must give their feedback and what type of further webinar they are expecting. They can also suggest that.
Yes.
So until unless we do not learn the technique in detail, we cannot analyze and we cannot interpret. Therefore, I request or I wish all the students take part actively in the webinar, and whatever the questions they have, they must put forward and move forward. And they can give their feedback and what type of webinars they are expecting in future also, they can give their comments. This is the suggestion from my side to all the participants. Thank you. Go on.
Thank you, sir, for putting up this point, because I know I'm trying to make it more interactive and everybody is welcome. It's not like just few of the faculties ask the question, because I can see there are many students. They are doing PhD, or some are doing their masters. I have seen few names. They are from their undergraduations also. So I want to emphasize on the fact what Dr. Venkat said, that this is one of the webinar that Dr. Rashmi asked me to do, that people have issues with the result interpretation of the GCMS data, and you get your data done with NIST, comparing your spectral lines with NIST library and whatsoever is there, and on that basis you publish. But I think you should know what is the fundamentals of result interpretation in GCMS, because machine does everything nowadays, but it is always good to have a human intervention in that. So still, if you all are having any question, please ask before moving into the result interpretation part. Or I will consider myself as the best teacher, that whatsoever I'm saying, everybody is registering in. No questions? Okay. So
As
now,
far as I know, Dr. Pranav, you'll get
yeah.
lot many questions in this section. Results
Yeah, okay.
and interpretation. They are waiting for that.
So for that reason, I am ready with my terms. So first, when you go for result interpretation, there are these terms which haunts you. What are data points? What is retention time? What is the spectra? What is the relative intensity? What is mass to charge ratio, or M by Z? Then what is base peak, ion peak, precursor ion, probability percentage? I think many of you got hit with this, right? So moving ahead, I would like to make things easier. So data point, here when you see a peak, at every corner of a peak, it indicates a RT value, a different retention time value. And each RT value have different mass spectra. So each RT value for a peak, for a chromatogram or ion chromatograph peak, is called data point. So at each RT value, you get a mass spectra, which can be some or means some time very similar to the one. Suppose if I'm taking this and this, both can have similar mass spectra, but there is a change in the base peak. Now, what is base peak? So when you see the mass spectra, you see one of the peak is at 100 value. Is at 100 value. 100 value means, suppose here, this is the relative intensity, 25, 50, 75, 100. This is what? This is percentile, not percentage. So there is a difference between percentage and percentile. So what do you see? That this 57 is having the highest intensity. And since it is having highest intensity, maybe it's not 100%. If it had been 100%, then how come these peaks are there? So definitely this is not the 100%. But it is the highest number of fragment. 57 mass to charge ratio has the highest number of fragments, and that is why we gave it as 100. We give it a place at 100 percentile. This is the topper, even if it got 57%, but this is the topper, right? Consider it that way. So this is at 100 percentile, and all other peaks are relative to this. Means if it is 100%, then whatsoever number of fragments 71 has is 50% to that. So suppose if we got total, consider total, we have 10,000 fragments that hit to the detector, and this 57 is like 570 hits, and this is the highest number. So we will consider it as the base peak. And the 71, suppose it got 285. So in comparison to this, if 570 is 100, then 285 is 50%. Similarly, they have their sizes according to the number of fragments that hit, right? So this is called base peak. Retention time, we already talked about it. A spectra. So when at a particular data point, when we break down the fragments, we see this kind of a spectra. This is called mass spectrum. This mass spectrum is on the x-axis, it is mass to charge ratio, and on y-axis, it is relative intensity. So when we see the relative intensity with mass to charge ratio, it is called mass spectrum. And so,
Sir, please repeat.
okay.
Sir,
So
please repeat.
this is the mass spectrum of a compound, wherein this is the base peak. So what is mass spectra? When there is a segregation of the fragments on the basis of their mass to charge ratio as per their relative intensity, then it is called mass spectra or mass spectrum. As you can see here, you can see how it is calculated. So this is 57. This is for C4H9 plus. Okay. This is 71. So you can see the spectral difference between these. The difference is because of there is a loss of a small ion here. So 71 minus 43, how much? 28. So there must be some ion which got broken down from here, from 71. So there you can see 28 or 29 here, one fragment. So on the basis of, but their intensity can be low. It's not like it can be like 100%. Their intensity can be low or high. But on the basis of their intensity and the segregation on the basis of their mass to charge ratio, they make a spectrum of fragments that is called mass spectra. Then the next one is base peak we talked, ion peak. Sometimes what happens that your molecular ion breaks down into a smaller fragment, and at the end you don't get to see your molecular ion peak. But in most of the occasions, you find your molecular ion peak. It can be very small or very tiny, and it should be very tiny, because if it has been broken down into something, then there is a possibility that your molecular ion is degenerated or have a very less number of fragments left that have not been broken down into smaller fragments. As you can see, 142, the half of it is 71. 142 divided by 2 is 71, right? So you can see how it is calculated. So there you see a peak of 113. The difference is 142 minus 113 is 29. So difference is because of C2H5. Where is C2H5? Here. You can also see the peak of C2H5 here. The another break is from 113 to 85. The break is of 28 units. So that is C2H4. It is ethene. So here it is very small, so you can't see ethene, but maybe that ethene would have broken into CH2, two CH2s. So you can see 14 here, right? So this is how you analyze your data. Now the third peak that you can see is 85 minus 57. So there is again a loss of 28. And there is no 28 peak. So it is possible that 28 is broken down into 14. So you can see 14. Right? So this is how you analyze your GCMS data. But I was talking about mass spectra. So you got it. You got the molecular ion peak. You already learned about precursor ion. Precursor ion is the biggest ion. That is going to be your molecular ion, which breaks down into a smaller fragment in later tandem mass separation, right? Like mass to mass or mass spectra with time of flight, it breaks down into further fragments. The last one is probability percentage. I got this question many a times. Even Dr. Rashmi, if you remember, you asked me about probability percentage. I can say, Rahul, that's true. When a pure compound is loaded on the column and you get the molecular ion peak, but it is not always true. It depends upon your ionization method. If your ionization method is robust, means you have used the ionization method that can break your each and every mole of your molecule into smaller fragment, then you are not going to get molecular ion peak. But then you can have your base peak and other peak fragments, through which you can analyze that, yes, this could be this particular molecular ion. But this is true that if you have a pure compound, the chances are that you get to see molecular ion, even if there is no, because the role of GCMS is to separate the mix. So if you have the pure, then you don't need to do GCMS, right? The purpose of GCMS is to separate. Chromatography का principle ही है to separate the components from a mix, right? So yeah, so you don't need to have a pure compound to run GCMS. If you have the pure compound that you don't need, then you don't have to run GCMS. You already know what compound you have. Then you can directly take pure compound and throw it for NMR, and you get the NMR data, and from there you can say, yeah, I have this compound. Then you don't have to go for, because GCMS is not a confirmatory, it is just a you can say a probationary technique wherein you can have a idea of what kind of compound you are looking for. So yeah, I was talking about probability percentage. So whatever probability percentage that you get after your GCMS analysis in your chart with the result, that doesn't suggest that this could be this compound, or there are the chances. It just say that there are many compounds, they have similar formula weight. They can have similar formula weight. So until and unless you compare the peak patterns, why I'm saying peak patterns? Every molecule or every compound has their signature peak patterns. They cannot change. Even if they have same formula weight, but they have different structural integrity, and so they have different peak patterns. They have different signatures. So probability can tell you by the GC or the total, by the total ion chromatogram. Just by seeing that, you can say, yeah, they can be methanol, or there can be different molecules having similar formula weight. But until and unless you go and see the mass spectra, you're not going to get the actual figure of what you are looking at. So let's talk about what does this retention time implicate. Wait a moment. Yeah, I got caught cold today because the weather was cold, and I didn't realize the weather changes here very frequently. So I didn't realize. I saw the temperature was high, and I just wearing that t-shirt, I went out and I got cold. But yeah. So when we say that what this retention time tells you. So GCMS data is a three-dimensional data. So here at x-axis you get M/Z ratio, with y you get intensity, and similarly in a different dimension, in a total ion chromatogram, you get to see the retention time with intensity. So the x-axis in this intensity retention time in total ion chromatogram, you see the retention time and the time from sample injection to the end of the GC run is retention time. So this can also be viewed as a scan number, which is the number of data points that I told you that whenever you look for different data points, they are different retention times. So when you click on different data points, you get different mass spectra values, and they are detected as a mass to charge ratio. So this is what we get to know from retention time as well. When you go for a two-dimensional chromatogram, which you say total ion chromatogram, in that you can collect all the values of the total ions in a, you cannot, those are not segregated yet. They are in a consortium, and they get segregated only when they pass through the mass analyzer, mass separator, and pass through mass analyzer and get means hit to the detector. And until and unless you reach to that stage, whatever analyte you get have all the peak, all the ions integrated within one what do you say, area under the peak. So all are under that. So two-dimensional chromatogram in like intensity versus scan or retention time is produced by summing the abundance of all the ions at a single data point and plotting it against the retention time. Like this is one curve and it has many data points, and under all these data points you can have all ions integrated as a single data. So this is more comparable to a chromatogram produced by a GC detector. However, you know that each data point in the total ion chromatogram is a separate mass spectrum. And then you get to this stage when each data point gives you a separate mass spectrum. An example mass spectrum of the straight chain hydrocarbon that is here, that is 142, the molecular ion size is 142, can be seen on the far right. And they have different spectral lines depending upon their fragmentation patterns and depending upon the base peak. However, there can be another molecule with 142 as the formula weight or the mass to charge ratio, but it is necessary, or it is essentially necessary, that it could not have 57 as the base peak, because their orientation, their structure, their everything else is going to be different. Their fragmentation is going to be different. So depending upon this, you can say, suppose this is your target ion, then you can say, while writing your figure caption, that this by seeing this mass spectra, you can see that the molecular ion is at 142, the base peak is 57, with the signature peak pattern of 43, 57, 71, 85, and 99, and 113 as the signature peak pattern. If you say this, then there is no question will be asked from the reviewer that how did you analyze that? Because this is what you have done is the maximum any chemist or any mass analyzer machine integrated with AI can do. So this is what you get the information, and this you cannot say by yourself. You will have to run a standard with it, or you will have to run and compare it with the NIST library. If there is something already there in the mass spectral collection, that is NIST library, wherein you have all kind of mass spectral data updated every day, depending upon the researches being conducted all around the world. They develop these spectral lines and they submit there for the reference. Whenever if you are running, you can have this. So in your yeah, Dr. Rashmi, please go ahead.
I have a question here, so that if we compare it with the NIST library, we don't need to run the standard. We can give the data as a in the paper, or
So if you have already run your data, means compared your spectral lines with the mass data available in NIST library, then yes, you don't need to run the standard.
okay.
But it should be near, the RT should be very close, like it's not should be very far. And you can, and it doesn't matter that other peak patterns are like, suppose 41 is big and 43 is small, that doesn't make much difference. But this should be there. It should not be like 43 is missing in this spectrum. Then you cannot say that it is the same compound. It could be different compound, right?
Okay.
Or,
So either we
or-
should, yeah.
or there could be one possibility that your 43 ion is so less and your technician has set the threshold value to so less, means higher, that your this ion peak is coming below that, then it will not be available in the spectra. So you will have to be very sure that you have to set the threshold to the minimum, so that whatsoever ion peak you get, suppose if there is 14 here, and it is set, the threshold is set to be like only if I get 50 fragments, then only it will count. Then what will happen
Okay.
if you are getting suppose in this case you are not getting 28 here, right? But you can see 28 unit missing from here, right? So
Hmm.
there is a possibility that you could have a 28 peak here. But since the
Hmm.
threshold value have been set high, and the number of like suppose I need minimum 100 or scan minimum 100 fragments and then read. So then what happened if it is 99 fragments for 28, then you are not going to see in the spectra. You can only see 28 mass to charge ratio fragment if it is above the threshold or the threshold have been set to 0. If you set threshold to 0, then you are going to see more noise. That is why we set to a certain limit like 50 or 100, and we compare the spectral patterns with the spectra that is available or with the standard that you have run. Yeah.
Okay.
Yeah. Rahul, you said something. I will have to open it. Where is it? Dr. Rashmi, can you read the comment that Rahul made?
Yeah, Rahul is asking that this graph is for fragments of the same long chain hydrocarbon compound, right?
Yes, yes. So this is the hydrocarbon, which is 142 plus 1, that is 143. And you can see this is why I said 143, because this is C4H9 plus. So everything that you see here is in one positive value, one positive charge. And so here, whatever you see, when you calculate the value, it is going to be 143 or 141, not exactly 142. I'm not sure. It could be 141 or 143, but it could not be 142, the one that you are seeing here. It is always either one addition or one subtraction of the value if the charge continues to be plus or minus 1. Because obviously, if a certain molecule is breaking, it is breaking into two ions, then one will be positive and the another will be negative, and both are going to have the value or the magnitude of 1 only. So the change in the actual molecule and the molecular ion peak that you get will be plus minus 1. So if it is 143, then you are going to get either 142 or 144. Okay. So we were talking about that RT value is not important in case of GC. Yes, it is not, but still RT value can be used for identification of target analytes with the standard that we run, that we have already talked about. But these RT values can also help you to understand the total ion intensity at a particular time when you know the target, and also when you can compare the mass spectral lines at that particular RT. Sometimes, suppose in your standard is coming at this 4.66, right? And the analyte is coming at suppose 4.92 or 4 point somewhere. It doesn't make much difference if their spectral patterns are same in your mass spectra. So you can consider it as RT1 is equal to RT2, depending upon your program that you have run, and also depending upon the column length. So sometimes what happens when you compare your spectral patterns with the NIST library, you don't see the same RT value there. But when you go to NIST library, there they mention about each and every GC and mass spectra, and there they say that what column length they use, what internal diameter of the column they use, what was the program they used. So you can try to reproduce the same kind of data if you use all the parameters that they have mentioned over there. So you can get the TIC peak exactly at the RT that they have mentioned if you are using the same parameters. Okay. Yeah, now we can go for So we already talked about TIC. So now we can take some examples. So this is for n-decane at RT 4.66. So also with this, Rahul, you were saying that we get similarity percentage or percent similarity in the output table of GCMS data. How much similarity percentage is considered significant? See, similarity percentage only tells you about that there is the possibility that these can be one of the analyte that you are looking for. But until and unless you compare the means it can be possible that if there is 0.1% similarity percentage, then still you can go for checking the spectra and you can get. So with GC, you cannot say that this is the exact analyte if it is saying similarity percent. So you will have to see the spectral lines and you have to compare with the which is already available in the NIST library, or you will have to analyze your compound with the standards. So depending upon that, it happens. And also similarity percent doesn't gives you a significant information about your compound. So I don't believe in similarity percentage or similarity means percent probability. When you go with Verizon, I have seen their software. They don't give similarity percentage. I don't know about now because I used Verizon like 5 or 6 years ago, and they don't give you similarity percentage. I have heard that only Thermo give you similarity percentage or probability, and also depending upon what kind of analytical software you are using. So most of the analytical software, they don't rely on similarity percentage or probability percentage. Okay, so let's take few examples now to understand how to analyze the data. We have already talked about that 142 molecular ion, 142 molecular ion that previously in a blue screen, but here it's a very simple example that we have n-decane at 4.66 RT. They have run a compound, but in that they got one n-decane at 4.66, and this is the total ion intensity. So this could be one data point. And in this data point, which come at 4.66, we are going to see the spectral lines. So you can say that, oh, this is the same one with a different example. This is C10H22 as the n-decane. The exact mass is 142.17, but you will see the mass of 142, not 17. This is exact mass, but when you calculate the formula mass, it could be 143 or 141. So you will have to check that. So 120 and 22, it's exactly 142. Yes. So here, as you can see, that they break down into different spectral fragments, with 57 as the highest. Total number of the base peak is the highest, and on this the relative abundance of other peaks are mentioned here. And on the basis of this, you can say that with the base peak of 57 and the ion peak of 142 and different peak patterns at 43, 71, and 85 and 99 and 113, we can say that this analyte is n-decane. This is the final verdict that you have given. The another example is of ethanol. So this is a very simple molecule, C2H5OH, or the chemical formula is C2H6O. Here the ionization is not M plus or M minus. This is a direct hit ionization. So it can be either ESI method, electron spray ionization method, or simple electron ionization method. Here it is not adding any charge. It is just breaking down the compound into two different fragments or different fragments. So the first one that you saw is 31, the base peak. So there is the possibility when you break C2H6O, it is going to break as, so 46 minus 31 is 17. Oh, sorry. 46 minus 31 is 15. So the difference can be of methyl group, CS3. So if you break down to C2H5OH as CS3CH2OH, then the chances of breaking bond is higher in case of carbon-carbon. And that is why you get CS3 as the highest, means CS3 as the maximum breakdown out of it. So the remaining is CH2OH, that is 31. So the most number of peak that you are going to get is either 31 or 15. Now, why 15 is so low? Because there is the possibility that CS3 have been further broken down into CH2 and hydrogen, or CH and two other hydrogens. That is why you can't see more 15, but you can see 31 because it has CH2OH. There is a carbon hydrogen bond which is more stronger than carbon-carbon bond because of electronegative, electropositive, means high electronegative and high electropositive atom. But in carbon-carbon, there are slight high, slight low, because both are carbon-carbon and both are at alpha position. So alpha position means they are from either of the side. If you count, they are going to be the first carbon. So either CH3 is the first carbon or the CH2OH wala carbon is the first carbon. So in either case, it is going to be alpha-alpha carbon. So between alpha-alpha carbon, the strength of the bond is not very high, very strong. So there is the possibility that it can easily break. But if there is oxygen involved, so there is less chances to break at the same energy or with the same energy. So that is why you get 31 as the highest peak and the rest lower. So again, from 31, you can see there can be hydrogen missing. So 2 hydrogen missing and you get 29 and so on. The fragment pattern is in this way. Here in CH3, CH2OH, there is the possibility that there is CH2 double bond CH2OH and 1 hydrogen is removed, and that is why that atom is ion fragment is of 45. So by this kind of estimation and this kind of breakdown, you can finally get to know that what could be the final molecular ion. If you don't know the target ion, it is in a full scan mode, then you can have this kind of understanding and you can detect which molecule you are looking for. The another example is a bit kind of, this was linear and this is cyclic. So when you see here, the peak ion is at 133. So to break it, there are chemical ionization method was also used. It seems like that. And when you use chemical ionization method, there is a possibility that you get one positive charge or one of a methyl from a methyl group. So you get, if the exact mass is 132, you are not going to get 132. You are going to get 133 plus 1. And it is called M+1 ionization method. So you get this. So here, as you can see, this is 1, 2, 3, 4 tetrahydronaphthalene. It breaks into 132 and 104. So it could be possible that one hydrogen from here broke down because it's a branch, so it became 132. And that is the possibility at all positions, from here, 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 from all positions. That's why we are getting 132 fragment is one of the signature fragment. But the biggest one is 104. The possibility is that it breaks the two cycle, and both the cycles are, both the circles are now, means both the benzene, this benzene ring is now separated with this cyclic pentane. So you are going to get 104. And with that, you can see different peak patterns. So here in this case, you can say that 104 as the base peak, 133 as the molecular ion peak, 132, and this at around 91 could be the signature peak pattern for this 1, 2, 3, 4 tetrahydronaphthalene. When you see this structure, this is more complex. This is in a chair form structure, and this is called chlordane. So in this, you can see the mass spectrum for chlordane having mass to charge ratio as a function of relative intensity of the normalized. When we say normalized, this means normalized with the highest peak, that is base peak. Normalized means the other peak patterns are relative to the base peak. So this is the normalized to the most abundant peak, that is 375. And then another one is 374, that is very close to it. So there are the possibility that one hydrogen from somewhere have broken down, and we got one 374. So there are the possibilities that 375 and 374 both are going to be the highest. Sometimes you can get 374 as the base peak, that could be possible, but then it could be a kind of isomer. So the question is, whenever we get 374 as the other peak patterns are same, but there is a change in just 374 as the base peak or 375 as the base peak, then there is a possibility that during the fragmentation and ionization, they change their structure. They could be, or you are running a racemic mixture. So in that scenario, you are going to get only in case if Dr. Rashmi asked that can two analytes have the same base peak. So it's only possible when they are isomers. Otherwise, no. And that too structural isomers, not the functional isomers. If they are functional isomers, they have different base peak. Okay. I don't know why I can't open the chat. So, Dr. Rashmi, can you read the last question
Yeah.
Rahul asked?
Yeah, Rahul is asking that, is it like if we are introducing a mixture of multiple linear and cyclic compounds to GCMS, then each of these compounds will be broken down and individual graph with multiple peaks will be produced for each compound. Because I am working on crude extract from ecological samples, I am not sure about the nature and number of compounds present in my sample.
Yeah. So when you are loading a mix where you don't know how many components are there, then you get total ion chromatogram first, right? And with each total ion chromatogram, there are different data points. In each peak, like I shown it here. So suppose in your sample, you have many compounds or many molecules, and when you run on GC, so you will find these kind of peaks, many peaks, and these all peaks have different data points for different RT values, and for each RT value you will get different mass spectra. So on that, on comparing the mass spectra with NIST library or with the standard that you have, you can say that this data point is relating to this particular compound. So yes, when you load different different compounds, you are going to get different RT values, and on each RT value with each data point, you will be having different spectral values, and with that spectral values you can detect different kind of. So whenever you do kind of soil sample, or when you take liquid sample from any pond and you detect for pollutants, you get this kind of data. You get a lot of peaks in TIC, total ion chromatogram, and from there you have to select each and every, or if you know that what you are looking for, suppose you are looking for any compound which is having suppose toluene, for an example. If you're using if you're looking for xylene or toluene as a pollutant in water, so you will get different peaks, and you will have to analyze each peak with all the data points that is possibly visible in your chromatogram, total ion chromatogram. Yeah. So here in this case, this is Claudine. In Claudine, you can see 37, 375 and 374. Any of them can be a base peak depending upon their orientation during the fragmentation. And E or S compounds or the racemic mixture, they can interchange their structure anytime. So that's not a big deal. So it is possible that you get in one sample you will get 375 as the base peak, and in another you get 374 as the base peak. But in that case, you will have to see other peak patterns. If they are falling in the same signature, if they are falling in the same pattern, then yes, this is the same compound with they are like isomers. So you can use that way. Analyze the data with that. The next one is cannabidiol. So it's a cannabis. This is from a diol alcohol from cannabis, and it is called cannabidiol. And the exact mass is 314. You can only guess that by getting the ion peak, but it is not possible that you are going to get the ion peak every time. So you will have to analyze the base peak and other peak patterns like this one. And sorry. Like 246, like 231, or like here 232. So here again, you are getting 231 and 232, but this 232 can be a base peak here because it is not intense. The relative intensity with 231 is very low. It is around 15 to 18%. So here, this is electron ionization mass spectra. So whatever you see 314, you are going to see the molecular ion as 314. So here, as you can see, the base peak is at 231. So whenever you are going to analyze a GCMS data, always remember that you have to look for three important things. The first thing is the base peak. The second thing is the molecular ion peak. If molecular ion peak is not there, then you have to see the signatures, the signature peaks. And the third thing is what mode you have used for ionization. If you have used ESI or EI mode, then you know you are going to get the molecular ion at the same place if you at the same mass to charge ratio, which is exactly equivalent to the formula weight. So these three things are very important to look for. Don't look for, I'm again repeating it, don't look for probability percentage that you get, don't look for percent similarity that you get. They are not going to help you. And it is going to be confusing because once you start, you will, I had one data from Waters. Waters is a company which makes analytical, chromatographic analytical systems like GCMS and HPLC. And in that, I got like 17 probabilities ranging between 60 to 70%. I was confused that how to analyze that data because it was the same thing that I was looking for. Oh, I got percentage probability. Like you do in bioinformatics, when you look for, you see that if E value is near to 0, then it is the exact thing. Or you see for the highest similarity, 99% similarity or something like that, then you say, okay, this genomic or this gene is the same one or homologous to the one. But here it is not going to help you. I thought that it is going to help when I was doing it, but then I found out that this is not the exact parameter to look for. So you always look for the base peak, the ion peak, that is molecular ion peak, and the peak pattern. And also the ionization method. If it is EI, ESI, then you are good to go with the same formula mass, that is mass to charge ratio. Now, this is very interesting. I'm looking, means I have been dealing with a paper few days ago, and they were talking about this gassing. This is called epoxy gassing, and in that epoxy outgassing, in that they ran a reference epoxy, and you can see here, and then they ran their sample. And in epoxy gassing, except epoxy, there should not be any other compound like benzene or anything. If epoxy gassing have, means if epoxides or epoxies having other organic compounds, then this is failed epoxy, you can say. These epoxies are something that you use uh, when you look for epoxy arts on YouTube. You will find that how people are using epoxides and epoxies, and they make different structures, different kind of molds, and they toys with that. So they need to be outgassed, otherwise they are not going to give you the proper mold. And for that reason, they tried the reference epoxy, and they tried with there, and they found out that they have benzene compound in it, as when they compared it with, so they have high benzene presence, and that is why they failed outgassing of epoxy. So this is the interesting paper that I came across, and I was reading. So you can see at 9, they have a large cluster of benzene compound. So by GCMS analysis, you can find these kind of data. But yeah, the question is, how did you get these compounds? How did you extracted this compound? Or how did you check it? Because if you are going to put it in GC, the whole sample, you are not going to get this, because epoxides get degenerated when they get heat. So then how they were identified? Rahul, do you have any method to get volatile compound without heating them or without burning them? If they are highly volatile, then how will you capture them?
Sir, I dissolve them in hexane.
What will happen?
It will separate the volatile compound from the air, and it will remain suspended. I'm not expert in this. I have just
No.
used.
I'm just asking you. I want to know the understanding that
Yes, sir.
how we can do that. If it is already volatile, we don't need to dissolve it in anything, right? You are making it more complicated, because in that case, if our compound is lighter than hexane in terms of weight, then you are going to see only the noise of hexane. You are not going to see. And if we set the threshold to like 100 or 500, then we are not going to see our compound. We are going to see only hexane, right?
Yes, sir.
The best way is there is a small setup that we use, and that is called CLSA, closed loop stripping apparatus. It has a charcoal surface on it, and charcoal absorb that volatile vapor from that particular compound. So when you put this epoxy and benzene, what will happen? It will absorb epoxy and benzene at a different absorption rate. Okay, and when you get that CLSA into that CLSA stripping apparatus, you hold that in front of GCMS near to that injector. What will happen? It will suck up the charcoal beads at which you have absorbed these vapors, and then charcoal have the capacity or the tendency to deal with high temperatures. So what will happen, even if there are high temperatures, the epoxies are not going to get degraded, because maximum temperature is uh, being bore by the charcoal, and these vapors are easily carried out with the carrier gas and reach to the detector. So then you can get these benzene compound and this epoxy compound separately visible, right? Because they have different masses. Epoxy is heavier than benzene, so definitely you are going to get separated when they are going to get separated when they pass through the um, tandem mass spectra, mass spectrometers, MSMS, right? The next one is oh sorry, the next one is vegetable oil. So there was yeah
Excuse me, Dr. Pranav.
yeah Please go ahead.
I think if the students are not asking questions, we should ask them to make sure if
I'm going to
Yeah.
ask, don't worry. I have the questions for them as well. I'm totally prepared. So that is why I picked Rahul, because he had shown, means he interacted and he had shown the potential that he have already worked. He is working with GCMS, so he must be having some information regarding that. That's why I asked him. And I want every other student to ask the question, especially
there are many
Art-
students like Varsha,
yeah, I
Shilpi,
can take all
Shalini,
names.
yeah,
Yes.
Sanika, and
Everybody,
Nishu.
please ask your questions. If you have any questions, you can ask. This is the best time. After that, if you're going to ask the question, you will have to wait for the answer, because I am not looking this form every day. I'm not going to see that form every day, the feedback form that I'm going to send. And also, I'm very much, oh, yeah, Dr. Sangeeta, please go ahead.
Yes, Dr. Pranav, I have a question.
Yeah, sure.
That is, what is the importance of running QC samples? Because when we were writing the paper, we were quite confused regarding the use of QC samples, and do we need to run it along with our main samples? So I was very not getting it, the point of the QC sample. So I wanted to know about it.
QC sample actually just help you. It's kind of positive control. It just help you
Okay.
to know that your program and your machine and your column, they are all fine. That's why you run QC sample. So
And
you can run it parallelly. Either you can run QC sample first and then run get the baseline and then clean the column and then run your sample. So you can do that parallelly. But I
And-
think yeah, go ahead.
one more point I have, that is regarding the preparation of the QC samples. Generally,
Mhm.
it is prepared as a mixture of all the samples we want to run.
Yeah. I don't think it's a good way to do that, because
Okay.
what I know that for QC sample, it is always good to run a similar compound, not the mixture. Suppose
Not the mixture.
yeah, for example, suppose I'm trying to run some terpenes. Okay, I'm looking for terpenes in my as an analyte, but I cannot run terpene as a QC, because if I'm going to run the same, suppose for example, I can say that take an example of terpene like germacredinol. Suppose I'm going to run, I'm going to detect germacredinol. So I will not run germacredinol as a QC sample, because it is going to give us the overlapping data, and that is not a good QC. For QC, you then I will run humulene. Humulene is a small molecular weight compound having similar structural organization as dermacredinol, and that I ran. So suppose you are looking for um, I'm taking an example of Dr. Rashmi Tripathi, is one of the compound hexadecane, suppose.
Okay.
If she is running hexadecane, right? And then I don't need to run hexadecane. I can run N-decane as a QC. That will be sufficient to give us the information about our program, our machine, and everything. Yes, the column
So
quality.
one more point I have, that
Yes.
is one is we were talking about the QC samples. Another point is that what are internal standards? We also use the internal standard, and generally we mix the internal standard compound with our main compound. So what is the significance of that, and how we, if we are running QC, do we need to run internal standards also?
Purpose is different. It is not the same purpose. When you do qualitative, then you run QC sample.
Okay.
And you can run QC sample for checking your column parameters and every other parameters that you're running with. So you can use QC samples with when you are going for quantitative analysis as well. But
Okay.
internal standard is basically used for quantifying the amount of
Okay.
quantifying the concentration of your analyte. For example, I know that 1 microliter of internal standard is equivalent to 1 microgram, right? So what will
Yes.
happen when an internal standard is something like that, like QC sample itself? Suppose I'm having my analyte is germacredinol, and I'm running humulene as the internal standard, and I have humulene in the concentration of 1 microgram per microliter, suppose. So what will happen, and they have similar kind of structure, both are terpenes, but they have different retention temperatures. They can't vaporize at the same temperature. So we should not, why we should not use QC as our samples, or why we cannot use our samples as a QC? Because it is going to give the RT at one of the analyte that we have, right? So we don't want any confusion. We want to resolve it. So for that, even in quantification stage, in the quantification phase, what we do, we take internal standard, which is similar in nature, like terpenoid, both are terpenoids, A and B, but I know that A comes up very very early in the mass spectra, right? It will come up like after 6 or 7 minutes, but our compound is going to come at 16 minutes or 18 minutes or so. So in that case, we know that they are not going to overlap. The peaks are not going to overlap. Then only we can quantify. Now what we have, we know that 1 microliter having 1 microgram of humulene, alpha humulene. So when now we get 2 peaks. One peak is for alpha humulene and the another peak is for our analyte. So now area under curve of both the peaks and taking the proportional ratios will give you the value. Suppose I have an area under curve is like suppose 10 centimeters square for alpha humulene and 50 centimeters square for germacredinol. Then we can say that we have 5 times more germacredinol in respect to humulene. So here the concentration is 1 microgram per microliter. There the concentration will be 5 microgram per microliter. So from here we can quantify. So this is the role of internal standard.
Okay.
But still
So
you can
that
run, yeah, but still you can run a QC sample for conditioning
For
and for analyzing the
for
conditions.
the
Yeah.
for seeing the conditions and for seeing the stability of the instrument and all.
Yes. Yes.
Okay.
Especially the
Okay.
column, because whenever you run your column at a higher temperature, what you see, you get kind of leaching of the silica, and
Yes.
you can see in the result you silioxane
Silica,
kind of.
yeah.
Yes, you get silioxanes and all. So these are just the
Yes, we
leaching
get
of
it.
your, these are just the degraded silica columns.
We get some silane compounds we detected. I
Yeah,
think that was from the silica.
that is the silica leaching. Yes.
Okay. Okay, Dr. Pranav, just I have one more question. That
Yeah,
is,
sure.
are we also going to discuss about the multivariate analysis for the GCMS data further in your lecture?
Not in this lecture, because if you are going for multivariate, it is going to be another 3 hours.
So, so this is my suggestion for the next webinar. Whenever we are proposing, you are proposing. That is, please also include the multivariate part.
Okay, I will try to include that. Yeah.
Okay. Okay. Thank you.
But I want to know, where are you going to use multivariate analysis?
Actually, we are working on a metabolic profile of
Mhm.
some tissue, and
Yeah.
for that, we are trying to put multivariate analysis.
So it is mainly the statistical part of it. So whenever we go for statistical analysis, or whenever we are going to include statistics, we are going to include that part. Yeah. Obviously, it is going to be whenever we do a stat, we are going to include that. Yes.
Okay. Thank you.
Yes, Dr. Pranab, there is a question for you.
Yeah. Yeah. Sure.
This is we load hexane extract of pheromones to the
Mhm.
GCMS column as suggested by the technician handling GCMS machine.
Yeah.
It is easy to pour hexane directly over the insect under study to prevent loss of pheromones. I was not aware about the closed loop stripping method.
Okay. For living insects, it is difficult to put them in closed loop stripping apparatus. And if you are talking about India, then in India, I rarely found any GCMS instrument having CLSA. And if your PI or if your institute can buy that CLSA, it's not a very costly thing. But just that in India, people are not working on any such kind of thing, wherein they can have directly capture the volatile vapors. That is why they are not using CLSA in India. So I have rarely come across any machine in India where we have CLSA attached to it, but it is not a costly affair. You can also make your own CLSA apparatus, right? Just like a charcoal coated loop through which you have to pass, means you have to have a sealed container containing charcoal coated loop, and that it just opened once when you are putting it on a, suppose I'm working on a particular bacteria in a Petri plate and they are growing in, I just throw a substrate in it and then cover it with CLSA. And we know that they are going to produce that product, which is highly volatile, and it is going to be captured with that CLSA loop. And then we can just take it off and put it back on the GCMS machine. So there is one modification in the inlet of the GC, wherein that CLSA fits very well without any, and they create that vacuum in it. So yeah, if there is a good engineer in your known, then you can get it designed and made. It is not that hard, difficult thing to do. Yeah.
That's all.
Otherwise, dissolving pheromones in hexane instead of hexane, I will suggest to go with more because go with more polar instead of going with hexane. Try pentane and DMSO to dissolve that, or not DMSO, pentane. I forgot that name. DMC, dimethyl chloride. Yeah. So use DMC and pentane in the ratio of 5:1, and I think it will give you better capture of volatiles. Okay. So yeah. Any more questions? So now those
No,
who
I
are not
don't think-
ask, now those who are not asking questions, this question is for them. So here, in this case, it's a food manufacturer who was using who was currently using this oil, the pattern that you are seeing on the top, and then they moved to the new oil having this profile, right? I'm going to take a name and then I'm going to ask the question. Dr. Rashmi, pick one. Oh, see, people started
Yeah,
leaving.
I'll do that. I'm waiting for the opportunity. Nishu Sharma, please unmute
Okay.
yourself.
Yeah, Nishu, please introduce what you are doing, and then we are going to have this question.
Uh, Good afternoon, everybody. I'm Dr. Nishu Sharma from PAU. I'm
Mhm.
research fellow working in a water project. Sir, actually, I
Okay.
have missed most of the presentation because of the network issues.
Uh-huh.
That's why I'm not asking anything. I have missed
Oh,
more than half of the presentation.
then the
Okay.
chances of asking
Yes.
more questions, then there is a chance of asking more questions because you already missed it. So you have a lot of questions. You should have, right?
Okay,
Yes,
then.
sir.
Okay.
Okay, next.
Damini, 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 please unmute yourself. I think Damini is either not there or having some problem. Okay, Krishna.
The next part of this presentation is very important, that how you are going to
Krishna.
present your data. So if you are going to leave just in a we are going to ask you the question, this is not going to help. Yeah,
This is not going to help them, actually.
yeah.
Actually, this
Krishna.
webinar is particularly for students and they are not actually asking the question. They should. And data
And ma'am,
representation is
it
very,
is,
very important. Right? Yeah.
ma'am, and this was only on their, means it was they who wanted this kind of webinar. So
Yes,
we have,
yes. They're
means I
not.
proposed because of them.
Yes.
Nishu, sir, I think I asked. Krishna, then Damini, Charu.
The name that you are taking, people are leaving. So don't take
Yeah,
names now.
I
I
don't know
think so.
what's the problem with them.
Leave it.
Charu, Krishna is, I think,
Good
unmuted.
afternoon.
Yes, Krishna.
Good afternoon, sir.
Good afternoon.
Sir, I want to know how to calculate NZ ratio.
You all are asking the
Okay.
same question and again, that is MZ ratio. You'll have to wait for that. First, we will move
Okay,
stepwise.
ma'am.
So
Okay,
first,
ma'am.
what we are asking,
Uh,
when you'll follow it in sequence, then you will understand things. From the beginning of the webinar, you are only MZ. So please answer the question first, and then we'll come to your question. Yes, Dr. Pranav, you were about to
Okay.
ask something.
Yeah, in this, Krishna, just listen to this question. It's not going to bite you. If you don't know the answer, it is fine. You can just say that I don't know the answer, or you can have a wild guess. It is not going to. Nobody here knows GCMS. Even I don't know GCMS much, but still, it might be possible that I could not be able to tell if your answer is correct or not. But still, let's try. So in this, this is a vegetable oil analysis that a food manufacturer was doing. So a food manufacturer was considering to switch from their current oil to a new oil. And when they analyzed their oils and on GCMS, they found out that their current oil contained several different compounds, which were, I can say that at 19 minutes, it was palmitic acid. At 20.6, it was linoleic acid. And at 20.7, it was stearic acid. So there are different retention times. Now, when they got the mass spectra, they got one thing at a same peak. That is, when they see the fragmentation, they found out that one thing is same in both, right? Can you find the first question is, can you find the base peak in both of them? Which one is the base peak? This one or this one? In the first one, and here it is clear. So this one or this one, which one is the base peak? Yes, Krishna.
The first one can be the base peak.
Here, this one? I tricked you. This is TIC, and we don't see base peak in TIC. This is total ion chromatogram. How we identified that this is total ion chromatogram? Because it has retention time on x-axis and intensity on y-axis. We can only see base peak when we are looking for mass spectra, right?
Yes,
So
sir.
this is the first trick that I played. The another thing is that since we don't, this is just the retention time graph with intensity, so we cannot say that what are the changes here. Only with this you can have two more compounds or three more fatty acids there, right? That's it. We cannot say what are what until and unless we are going to check the peak pattern. So see, such a simple question I was asking, and everybody is leaving that. Anyways, there is one residual solvent analysis. In this analysis, what we do and why we do, we all do residual solvent analysis, but we don't use it as a data. So why we do residual solvent analysis? Suppose there is a company which is manufacturing something, or suppose Dr. Rashmi is working on some compound and she is using ethanol or methanol as the solvent. But at the end of the production of that compound, or at the end of the production of that compound from the industry or the pharmaceutical company, it is expected that there will be no methanol. And we all know that methanol is poisonous, right? It's toxic to human consumption. So, but in the solvation process, we need methanol because there are compounds it doesn't dissolve in other, they dissolve well in methanol. So pharmaceutical companies use methanol as a solvent. So they use, but at the end, they wanted to check whether there is any methanol trace left in the final product. For that, they do residual solvent analysis. And in this, since ethanol, methanol, they are highly volatile, so what they do, suppose I made a syrup which has ethanol as a base solvent, I just open the lid and take the headspace and do the headspace analysis. So for headspace analysis, I just put the probe into the bottle, means from the top, not dipping into the liquid, just the headspace wherein just the air is there. So we just put the probe and have a negative suction. So what happened, whatsoever air is near to the interface of the liquid, it will get trapped. It will get sucked up into the needle in the probe, and then it passes through the whole process. It goes to the inlet, get heated up, get volatilized, passes with the carrier gas onto the column, and then again, depending upon their retention in the column, they move out and get detected, or they get broken down in different ion fragments, and then they get deflected in mass analyzer, and they finally hit the detector, and we get the peak patterns, right? So this is how that is done, and this is how we get the mass spectra of the residual solvent. So if there is any methanol left, even if the trace, they are going to be detected. And for that reason, we use high resolution mass spectrometry. So for any kind of pharmaceutical resolution or pharmaceutical residual solvent analysis, they use high resolution mass spectrometry with GC. Because there you cannot set the threshold to like 1000 so that any fragment getting below that is not detected. It is like, because you have to follow the industry standards, and it's not like suppose there is one company, Piramal Pharma, so they don't have that GCMS. they have to get their product checked at, like in US they have FDA. Similarly in India we have a different authority, different agencies who have those high resolution mass spectrometry based GCMS system. So they do that for them. And then accordingly, like, yeah, recently we got to know, yeah, FSSAI. Recently we got to know about MDH. So how they get that information that MDH has ethylene in it? Because ethylene is volatile when it come to in contact with air, it volatilizes easily, and so it is being detected by GCMS. And again, the headspace analysis was done on all those compounds. So you can see ethanol, and every compound has a specific retention position. So if you have the same column, same everything is same, then in that column you will see near to 2 minutes for ethanol. For ethylene, means bigger the compound, you see it farther in the retention, farther on the retention time scale. So this is it. Now we are going for the next part, how to present your GCMS data. So there are many ways people use, employ many
Dr.
techniques.
Pranav?
Yes.
Yeah, I would like you to please discuss M by Z ratio once again.
I will discuss M by Z ratio during the publication part.
Yeah,
Yeah.
thank you so much.
Although we have discussed M by Z ratio, but I don't remember who asked this question. Can you please tell me her name?
Krishna Kumari asked, and
Oh,
I
Krishna.
think many other students are also interested in this
Okay.
question.
So let's go with GCMS data. So this is my PhD data. I decided to show this here. So this is Verizon system that I used, and you see that in Verizon you don't get 100% or 175, 50. They give you the number of fragments that hit the detector. See, so the mass that my compound was 222. 222 was its fragment, its formula mass, 222.18 or something like that. And it on my machine, on that machine, it the RT was 10.36. So to represent your data, you will have to show your RT value. If you have just single, if you are targeting just the single peak, you analyze all and you just target the single, because your thesis has the target that you have to get only terpenes. So in that, I have a target that I have to find a terpene, and that was germacredinol. So comparing it with the NIST library, I found out that my came at this peak is at 10.36, and in NIST library it came at 10.39, just 0.03 seconds, or just 3 seconds was the difference. So, and the peak pattern then I followed, and it was like 59 as the base peak, then 67, 82, 93, and 107 were the different peaks. So I analyzed by breaking all the bigger fragments, like starting with 222, then I got 59 as the peak. So I had this structure, I removed this branch, I removed these branches, and then found out that this is the structure that is having 107. Then again, I broke double bonds, and then found out that this is going to make 93. And by this, I found out that this is the peak pattern that I was looking for. And when I when I compare with the NIST library, I find the same thing. And then I concluded that yes, the total ion chromatogram came at the, in total ion chromatogram, the RT value 10.36 corresponds to the mass spectra with their m by z ratio. Krishna, m by z ratio. I'm going to tell you about that. So the relative abundance of the ions with their mass to charge ratio is relatively, or is corresponding to, the RT values, that is 10.36. And when I compared it with NIST library, I found all the peak patterns. Although this was my thesis, so I didn't added all my peak patterns here in the caption, but it is always good to add the peak pattern in the caption. So I added, just after this, I had a whole paragraph on the peak patterns and the segregation of these fragments, what break from what and what I got. So I had that paragraph. That is why I'm not showing it here, but in a publication, you have to show this way. If it is just one mass spectra that you are going to look for, if you have many mass spectras corresponding to different RT values, then you will have to say A as this, or you have to select RT to which you are showing the mass spectra, and you have to put this plot inside this as an inset, like in here. Here, what is there? There is the spectrum, but you have to have the spectrum here and this as the inset here. So this will give a kind of view that at this RT you are having this spectra. So you don't have to put this total and chromatogram with in front of every mass spectra. Rather, in every mass spectra, you have to put this TIC with that particular RT highlighted in that mass spectra as an inset. Suppose if this mass spectra is for RT value something 8.8 minute, or this one. So instead of highlighting this, I would have highlighted this one either by putting a arrow like thing here to show that this RT is under consideration, and that TIC will be here if we have more than one RT values for which we are seeing the corresponding mass spectra. Does that make sense? I think it is kind of, I made a complex kind of statement, but I hope everybody could have got the material what I'm going to say, or what I was trying to. Anybody please confirm it that you got what I wanted to tell.
Sir, please repeat. I didn't understand.
Okay, so what you didn't understand? Tell me that first. Out of the whole, what you didn't understand?
Sir, what to
हिंदी में
sir,
बोलिए. It is not required that you have to frame your sentence in English. आप हिंदी में पूछिए, कोई बात नहीं.
sir, which graph is to be pasted in
Inset. In the inset. Okay. TIC. हम हर एक मास स्पेक्ट्रा के लिए ये जो TIC है, क्योंकि ये जो मास स्पेक्ट्रा है वो 10.36 RT का है. अब अगर हम मान लेते हैं कि 9.2 RT वाले का मास स्पेक्ट्रा दिखा रहे हैं, और हमारे पास बहुत सारे ऐसे मास स्पेक्ट्रा हैं, बिकॉज बहुत सारे RTs को हमें देखना है. जैसा कि राहुल जी ने बताया था कि उनके पास मिक्स्ड सैंपल्स होते हैं, तो उनके पास क्या होगा कि उनके पास बहुत सारे RT वैल्यूज होंगे, है ना, क्योंकि अलग अलग कंपाउंड्स हैं. अब बहुत सारे RT वैल्यूज हैं, तो बहुत सारे RT वैल्यूज के बहुत सारे मास स्पेक्ट्रा होंगे. अब हर जगह वो एक बार TIC दिखाएंगे और एक बार मास स्पेक्ट्रा दिखाएंगे, तो क्या होगा, उनका पब्लिकेशन जिस चीज थीसिस के लिए चल जाएगा, बट वो भी ठीक नहीं है. लेकिन जब आप पब्लिश करने जाओगे तो रिव्यूअर कहेगा कि भैया आप, या एडिटोरियल बोर्ड कह देगा कि आप इसमें 10 से ज्यादा फिगर्स नहीं लगा सकते. फिर आप क्या करोगे, तो डिफिकल्टी हो जाएगी. अदरवाइज क्या हो रहा है कि आप ये एक पेज में ही आपके 2 आ पाएंगे, उससे ज्यादा आ ही नहीं पाएंगे अगर आप दोनों लगा रहे हो. तो तो उस केस में आपको क्या करना है कि जो अगर आपके पास मल्टीपल मास स्पेक्ट्रा है तो उसमें उस पर्टिकुलर आरटी, जिस पर्टिकुलर आरटी का आप मास स्पेक्ट्रा दिखा रहे हो, उस आरटी को हाईलाइट करते हुए उसको आप इनसेट में दिखा दीजिए। ठीक है, अभी समझ में आ गई ये बात?
Okay, sir. Thank you. Thank you.
Okay. Rahul ji, whatever you are saying, if you look it at means very closely, if I can zoom it, it will be easier. Wait, I will try to zoom it. Uh-oh. Can you see the presentation? Anybody please confirm
Yeah.
if
Yeah, presentation is visible. Presentation is visible.
but the thing is, where did it
Yeah,
go?
although it's the values are
It
not
is
very
of the
clear.
similar size, not zoomed out.
Values are not very clear, but the graph is visible.
Okay. Damini, I will come to your question, but the first thing is that the problem is that I have to share it again because I can see that my presentation moved and it is not moving in the presentation that you are seeing. So I will have to run it again. Now is it visible to you all, the movement?
No, it's on the same slide.
Okay.
4.2.1. 4.21.
Okay, 4.21. So yeah, I will have to do it again.
एक चीज पता है? ये डिब्बे में काली माचिस थी, मैंने दूसरे डिब्बे से लाल माचिस डाली, वो नहीं जल रही। क्यों नहीं जल रही पता?
अब भी मूवमेंट दिख रहा है?
Yeah, it's moving. Yeah,
Okay,
it's moving.
Fine. So Rahul, as you can see, there is a very small molecular ion peak at 222, right? That is why I told you that if you set the threshold to be very high, then there are chances that you cannot see few peaks. For that reason, you have to put the threshold to low. And here I can see to make, because there were like too many fragments, as you can see the numbers is in billions. So I had to set the threshold to high so that I can only get these peaks. Otherwise it was going out of the range. That is why I had to put the threshold high. And that is why the 222 peak here is very, very tiny. It is like micro. Fine. Okay. What was your question, Damini? Can you unmute yourself and ask the question?
Yes, sir. Sir, how to calculate pole change? And sir, what formula log to? Sir, I don't understand about a log to formula using pole change calculation.
This is something which is out of the syllabus. But anyways,
No, sir.
for fold change, usually we do fold change only when we are doing quantitative analysis of GCMS. So if you are not doing quantitative, then there is no requirement of doing fold change. You can just have to find, you just have to show the detection part that we have identified this compound on the basis of the peak patterns. When you say fold change, this means you are talking about the quantitative part, and there it's always like there is a control and then there is a test. So when
Yes,
you
sir.
do analysis on control, and after the treatment or after any kind of induction, you see that there is more formation of that compound. Then if you have used internal standard, then by that you can just compare and you can say that with this much induction. Suppose I will take an example of a expression system. There is a expression system which is qmate inducible, that the amount of qmate I put, the expression increases accordingly. Suppose I was using 1x, and as I added 5x, the expression of that particular protein increases. This is not relevant
Yes,
to, this is just to tell you that how you can calculate the fold change. And for that reason, it is the thing I'm saying is not related to GC. It is related to qPCR. So, or quantitative PCR. In that, what I did, I checked the concentration of the expressed protein by increasing the qmate. And how I analyzed it, at 1x, it expressed the protein that I get quantified on qPCR. Then in another treatment, I added 10x of qmate, and I saw the next expression level. So in that, in qPCR, you see the CT values, and CT values by taking the CT value and normalizing it with the relative base value with the log 2, you get the fold change, right? So you can only,
Yes, sir.
for getting the fold change, you have to take log value of, what you say, log gives you the exponential change in their expression. So in GCMS, I have never come across the fold change formula, and I don't think it is relevant here if you are running the internal standard. And because this GCMS is not going to give you, this is just a predictive technique. You can only predict things. You can only get to know what kind of, if you already know the target compound, then you can use internal standard with the same, having same nature, having same characteristic, but different at the different RT values. And then you throw it in the solution mix that you have, and when you in the sample that you have, and when you run the GCMS, you automatically get to know that what concentration of internal standard you have run and what final product you are getting by the difference, by seeing the difference under the area under the curve. So I don't think fold change is required there. But yeah, you can
Sir,
say that
I
if
was
you are
already seeing the some research papers, the fold p-value and fold change in a table of GCMS
you
data.
are, so there is no table of GCMS data. You are talking about the statistical value of GC. When you go to
Yes, sir. Yes,
statistics,
sir. Statistical values.
yeah, so we will be doing it some other time, because for that I will have to show you
Okay,
with
sir.
data. I cannot just recite things. I will have to tell you with data, and then I will have to show you how to do that. Yes.
Okay, sir. Thank you, sir.
Yeah. Thank you.
Yes, sir. Statistical is entirely different thing. Actually, they are asking
Yeah.
about the values which has been, yeah, this has to be analyzed with separate softwares.
Yeah. So there are different
that is not-
methods by which we do, but for fold change, we obviously look for the observed values, and then we go for like I give you an example of CT values. So we are going to like normalize the CT value, like we do normalize the CT value. Similarly here, we will have to normalize the values with the base peak that we got, the relative intensity we have to check, and we will have to see all the concentrations, and then we have to go for that. So for that, we need a table, we need a data to discuss. We cannot discuss it by just telling it. So yeah. Okay. Any other question related to the understanding of how we are going to represent? So this is one way that I told you, that if you have only one RT value to show, then there is one way that you have to get the RT values and then the corresponding spectrum
may I
one
interrupt?
after another. Yeah, Dr. Jyoti. Yeah, please ask.
Yeah, sir. Yeah, sir, I have some small questions, queries.
Yeah, sure. Go
Like
ahead.
suppose for the plant samples, generally what we do some phytochemical analysis of the plant sample. So
Yes.
of the similar plant samples with different stress conditioning, Like
Mhm.
there may be some expression of other phytochemicals. Sometimes we get 30, sometimes we get 28. So is there any method of comparative analysis between the samples?
Sorry, I didn't get your question. Can you repeat that?
I mean,
Um,
I mean comparative analysis of the data which we obtained. Is
Mhm.
there, can we do some overlapping methods that the overlapping peaks can be omitted out, I mean deleted out, and the only peaks which are peculiar to the sample can be seen?
No, this is very unfortunate with GCMS data that
Okay.
with even with other techniques, qPCR, HPLC, you can do that, but with GC you can't. And that is why this is considered as more
Because
sophisticated
in
and authentic.
FTIR that we do.
Yeah,
In
you can do in
FTIR that we do.
even in
Yeah.
NMR you can do that.
Yeah.
With HPLC MS you can do that.
Yes, yes.
With HPLC you can do that, but once
Mhm.
the MS is there, you cannot do that because let me tell you how this works. So
Mm.
usually what happened when you have a conglomerate of two technique like selection separation technique and like chromatography and then detection technique like mass spectra, here it doesn't go like from GC to MS. The interpretation of the data is not through GC to MS, it is from MS to GC. You go reverse direction. How? Because when the analyte pass through the inlet, come to the transfer line, come to the column, and then come out of the column to the mass analyzer where it get bombarded with electrons and different chemicals like methyl, and it breaks down into different. So till there, you don't have control to have the idea of what compound you are analyzing, right? Once it
Yes,
hit
yes.
the, once it get segregated by different mass spectral, like either mass or time of flight or any other technique, it gets separated, the ions are separated and it hits the detector, it gives you the pattern, and that pattern then lays back to the data points. So it's not like data points give you the mass spectra. No, this is the very big confusion that everybody has, that we get GC, we get total ion chromatogram first and then we get mass spectra. No, actually we get mass spectra first and then the computer software builds it back to the total ion chromatogram. So this is not possible in GCMS to remove the overlapping peaks. Yes. Rather, you can
Okay.
add overlapping peaks, but you cannot remove it. It's very difficult. Yeah.
Okay, sir. Thank you.
Yeah. Yeah.
I
Yeah.
thought it is a part of some software analysis that the overlapping peak can be like seen with the values that can be manually done. Otherwise, there is no way.
Yeah, there are many softwares.
Mm.
There are many softwares. One is MS Nova that you can
Mhm.
use. Another one is just with the name Mass Spec that you can use.
Mhm.
But I think they are paid. There is one MDIS wherein you do the convolutions, and when you do the convolution, you can remove the noise, but it will still show you the values in the mass spectrum. It will still show you the mass spectral values. So, but it will
Okay.
definitely remove the noise, and you can have a better picture of what you are looking at. Yes.
Okay, sir. Fine. Thank you, sir.
Any more questions on this? So this is one way to represent your data. The another way is that if you have multiple RT values that you want to take, then you have to have your TIC in the inset with the same RT value highlighted for which you are showing the spectral lines. That is it. And in the captions, you have to tell about the base peak. It is not mandatory that you have to highlight it, but you like base peak is 59 and molecular ion peak is 222. No, you can say that suppose if this is one of the peak in the signature, the another one is 59. So you can write it as a sequence that suppose it is 58, so, or 55, then 55, 59, 67, 82, 93, 107. Then if any one of this is in the signature peak pattern in the NIST library, then you have to take that, and then this, and you can write in a sequence. That would be enough to show. Otherwise, what you can do, you can start with 55, then add 59 in the bracket base peak, then 67, 82 and all, then add 222 in bracket molecular ion peak. That could also do. So this is the way you can represent your data. This is one another way, that you will just take the screenshot of the TIC and mass spectra and you can put it. This is what you get from the system, right? And this is what you can get from different softwares. So this is from Mass Spec that I have used it. Not Mass Spec, this is from MS Nova. So from where I convoluted, because there were so many and I had to analyze all the peak, and from that I had to pick that one. So for that I used MS Nova for that. So you can do like this if you get the direct values. So you can just take this and you get these kind of structures. If you compare it with the NIST library, it will give you the most most suitable data that you get, and you can have this. So it will show you the RT values like 7.174 to 7.277, and it took for like this to this. So this is the data point where it is showing this mass spectra. Sometimes if you have only one spectrum that you want to show and you have a many peak and your purpose is to show the total ion intensity with respect to retention time, then you can show this kind of, but this is rarely used. Mostly we use like DC mass spectra with the inset if we have many mass spectral lines. Otherwise, if you have only one, then you can show it like A and B. So yeah. This is it. The summary, we talked about the introduction of the GC, of why GC is called hyphenated technique, and why GCMS is used for sophisticated analysis of volatile compounds. What are the method that can be used? What is the instrument design along with the working of GCMS? And then what are the detector and selection of the detectors? And the method is basically the ionization method that we already discussed about it. And then the result interpretation, how we are going to analyze the result. And the result interpretation is based mainly on three things. Who can tell me what are the three things that we have to consider? Damini, are you going to try that?
Sorry, sir.
What are the three considerations that we have to take for result interpretation of mass spectral data?
So, sir, MZ values and
Okay.
retention time,
Okay.
and sir, TIC.
Okay. Is there
Yes,
anyone
sir.
who can tell if Damini said the correct? And if Damini is not right, then can you tell me the correct answer? Anybody?
Krishna raised her hand.
Nishu Sharma want to say. Do you want to say,
Yes,
Nishu?
sir.
Yeah. What are the three
Yes, sir.
considerations?
Sir, base peak, percent probability,
Base peak
and
is
retention
correct.
time.
Okay. Anybody
Percent
else?
probability.
Krishna wrote base peak, ion peak, and what was the third one? I didn't get to see that. Peak pattern,
Peak
right?
pattern.
Perfect. Yes. So this is the correct answer. So for mass spectral analysis, we would like to see these three things. One is the base peak, another one is molecular ion peak, and the third one is peak pattern. But Krishna, if there is no molecular ion peak, then in that case, what we will do? Then what is the third consideration? That is the peak pattern. Signature peak is the peak pattern. So yeah, Kavita, that's true. But again, if we don't have the molecular ion peak, in that case, what is the third parameter to consider? Anybody else? If you have any idea, if not, just say you didn't follow that. Yeah. Mode of ionization. What is the mode of ionization? That is going to play a very big role if we don't have molecular ion peak, right? Then whatsoever we are going to get the signature pattern, obviously that is the most important thing along with the base peak. But if we know that the molecular ionization is M+1 or M+, then in that case, whatsoever be the corresponding values for molecular ion that we analyze with, that we estimate with the signature peak pattern, we are going to add +1 or -1, and we can say, yeah, this is the molecular ion peak. Rahul, you said something. Dr. Rashmi, can you read that, what Rahul said?
You're
Yeah,
saying
I
the
got it. I
retention-
got it. The retention time of base peak followed by other peaks following the base peak. Rahul, can you unmute yourself and tell me what do you mean by this statement? Because I didn't get any glimpse
No.
of that.
Sir, just I was trying to figure out the answer of your question. When molecular ion peak is not available, then what should we
No,
do?
that's
So
fine. So molecular ion is not there. That
Yeah.
means we are talking about mass spectra. And we don't see retention time in mass spectra. So,
Okay, sir.
so retention time of base peak. Oh, okay. So here there is a very big conceptual misunderstanding. So what happens? Let me take you back to this peak. So here, this is the chromatogram, total ion chromatogram, and for this data point, we have a retention time. And at this retention time, when we look for mass spectra, then we get this fragmentation pattern. For example, we get in here, right? So this spectral segregation is not, means each fragment has the same RT value here. So for this base peak, there will be no different RT value. Got my point? These patterns, this signature pattern is at the same retention time. And for each fragment, there are no different retention time. So with this retention time, 4.66 for N-decane, we have this spectral signature fragment pattern. Got my point, Rahul? Is there any confusion in this now? Because this is the most important thing to consider. So the first thing is the base peak. The second thing is molecular ion peak. The third thing is the signature peak pattern. But if there is no molecular ion peak, then we will have to consider the ionization method. If ionization method is ESI and it is going to ionize just making M+, then you will see whatever the breakdown you can do with all the kind of, like in this case, first this will remove. So this is CS3. So you are going to be getting 12 plus 3, 15. So there will be some 15 peak of 15 mass to charge. Then this and this will break. So this is now C2H5. it is going to be like 28 or 29. So you will get 29 peak. So with this, if it is m plus 1, then it is going to be whatsoever it is going to be 1 added in it. So instead of getting 15, you will get 16. Instead of getting 29, you will get 30. So this is how you will analyze. So that is why you have 3 things to consider: the mode of ionization, base peak, and the signature peak pattern. Okay, that's it. Any other question? Any other doubt? This is the time you can ask. Otherwise, it's 3 a.m. here. I have to sleep. Yeah. No questions? Dr. Rashmi, where is Saya and Varsha? Are they around?
Yeah, they have joined.
Okay. Then can I ask questions to them?
Yeah, sure. Why not?
Okay, Saya and Varsha both. We have
Yes, sir. Good afternoon,
this.
sir.
Good afternoon. Yeah. So we have this signature peak pattern of this compound. Okay. And I have said something about this, that if they are isomers, especially structural isomers or E and S stereoisomers, then base peak could be anything, 374 or 375, right? But if they, then how will you analyze this data? If suppose if it is 374 or 375 instead of 375, it is 374, then how will you analyze the data? What are the parameters that you will look for?
Sir, the base peak can never be same for the isomers, like you have said. So
It can be same. It can be same. It's not like for isomers, it can be same. Yes.
for isomers it can be same.
Yeah.
So we will look for base peak, ion peak, and peak pattern.
Right. But my question is something else. My question is, we have 2 kind of base peak, 374 and 375. By this, let me tell you one thing. Suppose this is 375 1 and the number of fragments is suppose 1000. Okay. The 370, sorry, the 374 is at suppose 900, right? Because they are very close. So the rest fragments are at just 100. If you add all the fragments, they are going to be just 100, right? So this can be, both can be used as base peak. But in this case, like where 375 is 100% and 374 is like 99%, then in that case, you are going to take it as a base peak. But still, the consideration is going to the peak pattern. The other peak pattern should be same. Even if the base peak can be 374 or 375, it is not going to affect other peak patterns, other peaks. Okay. So on that basis, you are going to analyze the peak pattern here and the mode of ionization. These 2 things you will have to consider here.
Yes. Okay.
Yes.
Ji. Thank you.
I don't see any further questions. So I kept this slide for you guys, but very few of you asked the questions. So yeah, and these are the resources for learning GCMS data interpretation. The one that I will suggest is there is a paper in Analytica Chimica Acta. This is automated supervised learning pipeline for non-targeted GCMS data analysis. Non-targeted means for? Who will answer? What is non-targeted GCMS analysis? In that, what kind of scan you do? Full scan or targeted scan?
So full scan. I mean, it's random kind of.
Yeah. So this is the paper for non-targeted. So you have to do the full scan, and That is what they have talked about, and it is a very good paper. But the one that I followed to learn mass spectrometry analysis, that is interpretation of mass spectra from McLafferty, F. McLafferty. So I will have to find this book. If you guys need, I will send you the soft copy of this book I have. Otherwise, you can also go with spectrometric interpretation of organic compounds. They're very old books. It was first published, and the third edition was in 1980, and after that it was not published. So we have the old method, but I think that method is very much, because nowadays we have softwares, we have Python supported softwares, R supported softwares, but I think the method that they have taught in this book is going to keep you like ahead of these softwares. So this is a good book, Interpretation of Mass Spectra, third edition by Farhan McLafferty. So this is a good book. So I have this book. I will have to look for. I tried looking for this book. I didn't get much time to do that. Even I will tell you that I made this presentation in just 3 hours. I didn't have much time to look into more. I wanted to add more case studies to go with, so that you all could have got more experience with this. But I think the different kind of case studies I put in, that would be sufficient to start with. And obviously you will have to start analyzing your data. If you have your data, analyze it, and if you get any question, then there is a form. It's a feedback form with a section for adding your questions. So you can add your question there and let me know if you have many questions on that. Then I can just return back whenever I get time. Yeah. So I have pinned this link. You all can go, click on the link, go to the feedback form, fill in your experience whatsoever you have, good, bad. Be as critical as you can be, because it is going to help me better my way of presenting things, improve my way of delivering these kind of webinars. So yes, you can be as much critical as you can be. You can talk about my method, you can talk about my language, whatever you like. You can feel free to talk about it. And also you can put forth your questions whatsoever, and I will try to come back to you through email. Yes. So the last thing is feedback. So if you all need, just get a screenshot of this. Otherwise you will all get the certificate and the recording. So always you can find these resources. Yeah. So I'm really very thankful for getting this opportunity. Dr. Rashmi requested me to do this webinar, and I'm happy that even if I didn't have time, I had to manage a lot, but still, after all this, I was able to manage and get this done. So I would like to thank Dr. Rashmi for making me do it. This is just a beginning, but yeah, you kind of not only requested, you kind of forced me to, you should do this because you know this and people are willing to learn this technique and learn how to interpret the data, and you forcefully made me do this webinar. So thank you, Dr. Rashmi. Thank you, Dr. Venkat sir, for being the integral part of this thought process. And also, you have been my great mentor every time, whenever I looked for any suggestion or any kind of feedback, you was always there, and obviously unquestionable support that I got from you. So thank you for being that pillar of my life. Every time when I look behind, I see that there is someone who is um, patting my back. That go ahead, I'm shadowing you. So yeah, um, thank you, sir, and thank you everyone, especially Dr. Sangeeta, Dr. Jyoti, and Rahul for asking questions, which um, give me the opportunity to answer yours, and maybe I would have been able to quench your queries. But if you have any further questions, please come ahead and write in that feedback form. There I left a question. There I left a section for questions, so you can always put in there. Yeah. Yeah.
Thank you, Dr. Pranav. It just came into mind when the students requested so much, and it was very
Yeah.
kind of you to do this at such an odd time, odd hour.
No, that's
But
fine.
you,
I always, I always used to be awake
yeah,
at this time. I used to be in lab. Yeah.
yeah. But when students requested, somewhere I had the confidence that you will not say no to me, at least.
Oh, yeah.
So, yeah, so that confidence made me move ahead. So I really thank you, and it was a wonderful webinar. And you took up questions and means explained it in a way in which the students can understand it. And the entire time was utilized just in the explanation. That was the best part of it. So I really thank you a lot. And I think Jyoti ma'am wanted to share something. So Jyoti ma'am, if you would like to say
Yeah,
something,
sure.
please.
Yeah.
Yes. Sir, I also want to thank you. I'm also faculty of Banasli Vidyapeeth. And on behalf of all Department of Bioscience and Biotechnology, in fact, Rashmi, I am thankful to the Rashmi also because Dr. Rashmi has arranged such a wonderful webinar, and more participants should be there. Rashmi ma'am, if you can arrange, we can arrange some same type of webinar during working, not in the holidays time. Like
No,
July
ma'am,
onwards, we can also have.
ma'am, actually, holidays were the perfect time because we were having, we were not busy with the classes, so we could make it
Yeah.
out. And
No,
I
no,
think
I'm
for
saying
research,
because the students are not there, we are having more than 100 students in the department. So it
Mhm.
would have been more fruitful, but it's still, I'm thankful to the sir, Pranav sir, because it was so in-depth knowledge about GCMS. He started with the general principle and main working, how it works, how the analysis can be done. Everything was perfectly explained. And I'm very, very thankful to both the organizer, Rashmi ma'am, as well as Pranav sir.
thank you, ma'am. And
Thank you, Dr.
Dr. Tripti, would you
Jyoti.
like to say something? Tripti ji, would you like to say something? Share your feedback.
Yeah, just give me a minute. No, I'm there. Just give me a minute because I have logged in from my mobile. Am I audible to you?
Yeah. You're quite audible.
Yeah,
Yeah.
you are audible.
Yeah. Thank you, Dr. Pranav. I have been there since the beginning. Though my colleagues, Dr. Rashmi, Dr. Jyoti, Dr. Sangeeta would understand that we have gotten off, so it was a bit tricky. But still, I had been there with you since the beginning. I had certain questions because I am also delving into GCMS analysis at the moment. I was in the middle of typing my question with you. However, whatever my questions were in my mind, I discussed with Dr. Rashmi also yesterday before to joining your webinar. Result interpretation part was discussed very nicely. I am extracting the phytoconstituents of my bryophytes both in methanol as well as ethanol. Actually, one of the reviewer asked to use methanol as well because you rightly pointed out that methanol is toxic, and also ethanol has higher dielectric constant, so the
Yeah.
extraction would be better, right?
Yes.
So
Yes.
that part also you dealt very well because I had that point in my mind, and also the overlapping peaks.
Yeah.
So I think, whatever doubts I had in my mind from the beginning, they were sequentially and eventually dealt with in your presentation. And that's why I kept on going with the flow. And I really liked it. Thank you for being so informative. I think it will be helpful for us to interpret the result, not only for our thesis, but also for the publication part of it. Because
Yeah.
eventually that is the thesis is one part, and the publication is the part that goes on even after you have, completed your thesis.
Yeah. Right.
So,
True.
thank you so much for putting up such an effort for in such a short span of time. I think we and all the students appreciate it a lot. That means a lot. Thank you, Dr.
Um,
Pranav.
Thank you for your kind words. I was able to manage and put forth whatsoever information I had, whatsoever knowledge I have gathered during my PhD for working with actinobacteria, synthesizing secondary metabolites from it, especially terpenoids. And whatsoever information I got during that time, I think you learn always when you are in scarcity. So I learned in that situation. So and for that reason I always try to, and when Dr. Rashmi said that students are asking, since I had already dealt a lot during my PhD, so I thought it's a good scenario to come up with. And she knows that I never say no to students, so and I am very my motive is always to be helpful to students in whatsoever they want to get through. So still, if students have any question, they can come up anytime. They can fill in the feedback form. But I will tell you that feedback form is not for a very long time. It is going to be till 30th of May, and after that, I will discontinue that. So please write your queries whatsoever you have in that feedback form as soon as possible, so that, or while interpreting your data, GCMS data, and write your questions so that I can come up with the apt answer to your queries. And yeah, thank you for your kind words.
Thank you so much, Dr. Pranav. That is true.
Yeah.
The more you experience, the more you're able to speak. Thank you so
Yeah.
much.
Thank you.
So thank you all the participants. So I think we can leave now.
Yeah, if you don't have any,
Yeah.
if you guys have no queries, then yeah. Otherwise, I can speak more on GCMS if anybody is there to talk to me with.
I think, sir, it's time for you to get sleep. You
Yeah.
No,
have
it's the
already
time for you to wake up actually. It's already 4 a.m. there.
No,
Yes.
you know, Dr. Rashmi, that I sometimes I come
You
at 3 a.m. From my lab,
from the
so
lab. Yes,
that's,
yes, yes.
yeah.
I
So that's
understand.
not, yeah.
Yeah,
I
this is
come
why I
early.
was confident.
You,
This
you.
is why I was confident that you will not have any problem.
I told you that I am very scared
Um,
because I had not prepared my presentation. I just had my outlines, and I prepared when I came from lab today at around 5:30 in the evening, and then I started making this presentation. And by that time, it was just 6 slides, and then finally it ended up with like 41 slides, I guess. So I'm really, really happy that I was able to do it. Even if it's not a good presentation, I still believe I could have added more information in it. But I think for now, for those who are new in this area, they can have more to explore, and they can have a baseline from where to start. So I think with this baseline, they could
No, I think
make a high peak.
I think too much data would have confused them. So
Yeah.
whatever
No,
it was sufficient from them to start.
My thought was to add more case studies, different peak, different RTs and different peaks and all those things to include, so that they can have variations in that. But I think this baseline has been set now, so you can now reach the peak of that interpretation of the data. And this is how GC works. So just maintain your baseline and try to get the peak of whatever you are doing. Yeah, this is the motto of GCMS webinar, I guess. Yeah, thank you all. Yeah.